The meeting ID is invalid. Please re-enter your meeting ID followed by the pound sign.
Can everyone on Zoom hear us? Hey everyone, welcome to day 2 of your revision lectures run by MUMIS. Like yesterday, my name is still Hriday Patel and I'm a year 2 ACREP. Ramod, the pre-clin VP, has organized these sessions. I'm just presenting them today because he's unavailable. And we're going to get straight into it because we don't have much time. I'm going to put snacks on one of the front tables. We've got even less than yesterday. The budget was even less than I thought it was. So feel free to grab them and we'll get into it with biochemistry. Enjoy.
Hello. My name is Isabel. I'll be taking you through the biochemistry for Sem 1. It's a lot, so just be prepared. I won't go through everything in insane detail, but all of it's on the slides if you want to have a look in your own time. But with biochem, I think the biggest thing that you should take away is just like get the whole picture. Don't get too caught up on like the little details. Cool. Okay, so this is kind of how we're going to run through things. So starting with like molecular biology of the cell, going to central dogma, genes, cellular energetics, metabolism pathways, and then specifically alcohol metabolism, and then lastly finish up with like the cancer bit of the biochemistry. Okay. So first up is just what DNA is, its structure, etc. So with this one here, there's kind of just a couple of things I want you to take away from it. Okay, so just focusing on this diagram here. So we have like obviously 4 different bases here. You should know adenine pairs with thymine, cytosine pairs with guanine, and they are joined together by hydrogen bonds, forming this anti-parallel double helix structure. And then just how it kind of rolls around these histones here. These histones roll around each other. A group of 8 of them forms a nucleosome. They just get more and more compressed, form chromatin, and then eventually form your chromosomes there as well. And just with DNA as well, just know that everything, most of it is synthesized from a 5' to 3' direction, meaning everything is read in a 3' to 5' way. So you can remember that like 3, 4, 5, that's the order you read things in. The only exception to that is messenger RNA, which is read from the 5-methyl cap to the 3' by the ribosomes. Cool, okay. So we have replication of DNA, really important to know. Hopefully most of you did biology know this already, but just going through it, just to recap, so you have like 3 phases of this. So first, initiation. So pretty much where, you know, the acetylation removes positive charges on the histones, which forms euchromatin. So euchromatin is the one that you can actually replicate. And you can see here that at specific origins, DNA replication complexes attach. They have all these replication enzymes gets recruited with it. And so you can see the first step here. We have topoisomerase, which pretty much uncoils the DNA. So you can see how it's like in that really formed double helix, and now it's kind of getting uncoiled. And then we have helicase kind of breaking apart all the hydrogen bonds. And then the single strand binding proteins, which pretty much stop the bases from pairing together again. So that all is the initiation phase. So next you have elongation. So you have here your DNA primase, which is kind of where it starts, which attaches these RNA primers here. From there, your DNA polymerase, you can see here, will synthesize new DNA bases, attaching them in that direction there. So you should also know we have your leading and lagging strand. Leading strand goes towards the replication fork. So you can keep going because it's opening in that direction, while your lagging strand goes the other way, simply because of, as I said, everything has to be read in that 3' to 5' way. So the lagging strand is synthesized like discontinuously. You have multiple primers, and hence you have these, what they call also Okazaki fragments. And then after that, you have your termination phase. So this is the last phase of DNA replication. So your RNA primers are degraded because they have to be replaced by DNA. And then in your lagging strand, the Okazaki fragments are joined together by ligase. That's pretty much the glue, DNA ligase. And pretty much from there, that's the extent of DNA replication that you have to know. One small thing on top of that is we have, it's not working. Okay. Telomerase action, which maybe is a bit different to what you learned in high school. But basically what this one means is it just prevents the ends of the chromosome from getting degraded. So you can see here that we have this telomerase, which then attaches to the end of the DNA on the 3' end, synthesizing it in this direction. So as you can see, the DNA is now longer than it needs to be. So on the lagging strand, we can start where the telomerase has this little action there, and it can synthesize back in this way so you don't lose the end of your chromosome. But as you know, like this kind of doesn't entirely protect the chromosome. So as you get through life, your ends of your chromosomes get shorter and shorter. And that's basically because of this whole mechanism here. Okay, so just some things about prokaryotic versus eukaryotic replication. I won't go through this now, but just is here if you need it. A lot of these slides have been adapted from PS Pushing, the PSP from last year. So if you want to look more at that, you can have a look at their slides as well. Highly recommend. Okay, once again, we just have, key notes here. So DNA damage, types of damage that can occur. So deamination, pretty much the C turns into a U. That's pretty much all you have to know. Pretty much messes up all the structure of the DNA because now it's has this U here. And then you have bulky lesions. So instead of attaching across the two strands, they kind of attach next to each other. So adjacent bases are becoming attached. And then finally, double-stranded breaks basically just cuts down the middle of the DNA there. And then there's either two ways you can rejoin that. You can either delete some of the DNA and then join it back up, or you can use like the sister chromatid as a template, which kind of protects the DNA more. Okay, and then again, I won't go through this, but just this is quite important to know. So not only important to know what types of error minimization there are, but it's also important to know when it happens. So you have one during elongation, during replication, after replication, and throughout cell life. So this is very high yield, but yeah, it's not much to explain. You just kind of have to remember why, remember what happens and when it happens as well. And then, obviously the mutation is quite low, the rate here. So obviously it has to be quite low because they can be dangerous. You want to kind of protect the integrity of DNA. Okay, so now we have pre-transcriptional regulation. So this is when you're making proteins, you need to transcribe them, followed by translation. So I've kind of color-coded here the ones which promote transcription, and the red ones there, the ones that kind of inhibit transcription. So we can see here acetylation, good thing, accelerates transcription, methylation inhibits, transcription factors promote transcription. Activators, as the name suggests, also promotes transcription. Repressors, as it also suggests, turns off transcription. So it's important to kind of understand how these work to an extent, but I guess it's more important to know the overall effect of what these do. And then simultaneous control, once again, it promotes transcription and transcription factor regulation can have like a variety of effects, which you can see here. So not that important, but the ones on the first couple of slides are the ones you should be looking out for. Okay, and then just a bit about RNA structure. So you should know that there's 3 types of RNA. So you have your messenger RNA, ribosomal RNA, transfer RNA. So what messenger RNA does, that's the one that is being synthesized from your DNA. That's the one that's being read from your ribosome. So this one here is pretty much the blueprint for making your proteins. While your ribosomal RNA is pretty much making up the ribosome. So your ribosome comprises of different segments of rRNA. And finally, transfer RNA, it's the teleport, it's a transportation system. It brings the amino acids to the ribosome in the complementary base pairing rule with the codons on the mRNA. Okay, so we have different steps of transcription as well. So initiation, elongation, termination. So these names should kind of guide you as to what actually happens. So you have a promoter region on the DNA here, which kind of brings along this RNA polymerase, which is big green blob here. And then we have an initiation site, which is where the transcription starts. So as you can see in the second one in the elongation here, that this RNA polymerase starts going along here, transcribing the pre-mRNA using free mRNA nucleotides, and then it pretty much just continues on throughout until the end codon is reached. So at this termination site, transcription ceases, and then RNA polymerase gets released, pre-mRNA gets released. So note how I said pre-mRNA. So it's not done yet, still needs a bit of processing in order to get turned into a protein. So we can see here there's different post-transcriptional regulation. So remember before we had the pre-transcriptional regulation, we then went and did transcription. Now we have the post-transcriptional regulation. Okay, so we can see here there's a couple of ways that we can do it. So alternative splicing. So you should know that your DNA has introns and extrons. So your exons are the ones that are spliced together. So you can see here in this diagram, we have these segments of exons. Those ones are spliced together, so the introns are spliced out. And we can see here, we can include different variations of those exons to get completely different synthesis of proteins. Then we have pre-mRNA modification. So mainly what that is you have the methyl G cap at the 5' end, poly A tail on the 3' end. Just pretty much helps with transporting it as well as being read by the ribosome. And then we have RNA silencing. Probably not as important, but you can see a kind of diagram here as to what happens. So it gets folded, gets diced, and pretty much just binds to other mRNA, which inhibits translation. So when it has this, micro mRNA on it, it inhibits translation. Okay. So it's kind of important tRNA structure, but basically just know that it's a 3-dimensional shape. It attaches to a codon there with the anticodon on the tRNA. And then we also need to activate it to actually attach the amino acid to it. So that is done using tRNA synthase. Ribosome structure, also quite important. basically small subunit, large subunit. And basically on top of that, we also have 3 sites. So pretty much goes from APE. So that's kind of the order that the tRNA binds to the ribosome. So we have the amino acid site, peptidal site, exit site. So that should kind of give you an indication as to what's happening. So amino acid site, that's where the charged tRNA with the amino acid attaches to the mRNA codon. Peptidal, because that one there, it's growing the polypeptide chain. That makes sense. Exit sites, where it leaves. So very self-explanatory. Okay, so we have now different also phases of translation. So it's very similar in the sense of what actual names these steps have, but we can see here that we have translation initiation. So first, it binds to the small subunit of the mRNA, and it goes through, it's bound to the 5' methyl G cap. So that's where it kind of starts mRNA reading. But the actual synthesis of the polypeptide chain won't start until you reach the start codon here, which is AUG. And then that's always the first amino acid of all proteins. And then from there, once that first tRNA attaches, we have the large subunit attaching there as well, which then, kind of brings everything together. So next one there, as I said, this one just kind of follows the kind of processes of the different spaces in the ribosome. So the APE pretty much just follows that thing around. The 2 subunits, C, as you can see in this third picture here, kind of slide along to move it, and then replaces itself there. So just know the order of how it goes through the ribosome. And then finally, termination. So once you reach a stop codon, there's no tRNA recognition. Instead, a release factor binds to it, as you can see this grey blob here, which kind of makes the ribosome tear apart, and then the mRNA can also be released. Okay, so just like we had post-transcription regulation, we also have post-translational regulation. So this one here is probably not as important as the post-transcriptional regulation, but it's good to get your head around. So pretty much target localization. So telling the polypeptide where to go. Do you stay in the cell? Do you need to leave? Do you have to get transported somewhere else? And then you also have chemical modifications to these. So these ones are quite self-explanatory given the name. So phosphorylate, add phosphate, glycylation, add sugar, proteolysis, protein lysis breakdown. So it's folding and kind of removing parts of the different peptide. And the last one here, ubiquitination. So ubiquitin protein coupled, and pretty much it just marks it for breakdown. There's more here. I won't go through it. You can read that in your own time. Okay, questions. So these questions may or may not have been covered in what I just said, but whatever I don't cover, you'll learn it now. So the first one there, within the structure of DNA, we have 4 answer options. Someone from this table over here want to give me an answer? C, yes, it is. So we should know here that we have 2 pairs of bases. So we have pyrimidines and we also have purines. So we have C there as the correct answer. So we should know cytosine and thymine bases are pyrimidines, and that leaves adenosine and guanine bases to be purines. How you can remember that is adenine, guanine, like all good, so they're pure, purines, which leaves cytosine and thymine to be pyrimidines. So why the rest of them are wrong? So A, there are actually 3 hydrogen bonds between cytosine and guanine. As I said, I explained B. And D, nitrogenous bases are bound in the complementary base pairing by hydrogen bonds. Cool, okay. Next one there, during DNA replication, DNA ligase, what's its role? Someone from this table want to give me an answer? Yes, that's correct. So A is the correct answer for that one there. So it joins the gaps created by the removal of primers and addition of replacement nucleotides on the lagging strand there as well. Yep, lovely. Okay. Moving on to the next one then. Okay, so we have there, which of the following correctly pairs the correct nucleic acid with its function? Does anyone want to shout out an answer? C. So we have tRNA there, brings amino acids to the ribosome, synthesizes that polypeptide chain. Lovely, lovely. Okay. Okay, during the transcription of DNA to synthesize RNA, table in front of me here. Can you give me an answer? Yeah, C. So energy is supplied from the hydrolysis of nucleotide triphosphates. So hopefully you just know the rest of them are wrong as well. So DNA helicase breaks apart the DNA double helix. RNA polymerase is the one that kind of, creates that RNA primer. It's actually not involved in DNA transcription as much. And then, RNA polymerase, again, is not really involved in, most synthesized RNA. Oh, so this one here is, again, it reads 3' to 5'. So remember, it always reads 3, 4, 5. Okay, next one. During the transcription of DNA, we have that already, so C. Which of the following statements is true regarding the production of mature mRNA from pre-mRNA? Someone from that table there want to give you an answer? Shout out. Did I hear D? Yes. Okay, so yes, D is the correct answer there. So we should know there that your polyadenine tail is added to the 3' end of the pre-mRNA. That means the methylguanine cap is added to the 5' end. Cool. Okay, that brings us to the end of all that DNA, RNA stuff. Questions about that?
Any questions?
Okay, easy. Okay, so let's speed through this. We gotta get through this. Oh, question? No.
Thanks. This is a question about the GCAP. I think we've also heard that it's the site where initiation occurs during translation.
Yeah.
But then, so I'm just a bit confused about that. Does it have like 2 roles?
2 what, sorry?
2 roles, 2 functions.
Well, they have different functions. So its main function is obviously like transportation, but it's also that recognition which the ribosome can start reading the mRNA. So it starts reading from the methyl GCAP end. Oh, okay. I'll stick through it. Yeah. Okay, next, genes. We're gonna speed through this. This is just reference slide. Can do that. Okay, questions. What is the use of the Harvey-Weinberg equilibrium? If I get attention, please. Okay. Someone want to shout an answer? Quick. A and B, trick. Okay, so they predict genotype frequencies from allele frequencies and also try to see how evolution occurs well by how the alleles change. Okay, next one. What is heterozygote advantage? Okay, sorry. So there may be a third factor as to why being a heterozygote is advantageous to survival, hence increasing the proportion of heterozygotes. So we know that there with you learned the one I think malaria, and that is that third factor there as to why being a heterozygote is advantageous. What is not a disruptor of the Harvey-Weinberg equilibrium? Yes, good. So you actually need random mating. Lovely. Okay, next one. This one I won't go through as much. You can read through it, but important to know the different types of biotechnology. So you have farming, therapeutic cloning, gene editing as well. Mostly knowing CRISPR-Cas9 is important, how that works. And then also recombinant DNA. So it's important to know how it works here, so the different steps. Also important to know why certain host cells are used. So why mammalian cells are used, because they can handle post-transcriptional modifications and larger proteins. Yeast for speed, and bacteria because it's cheap. So know those advantages there. Also know the polymerase chain reaction is important. So what are the requirements for it and the 3 steps? Basically just know it goes from high temperature, low temperature, high temperature again, breaks it apart, adds primers, extends the primers. Very simple. And then just know a bit about somatic cell nuclear transfer as well, and kind of know the legality around it as well. But again, it's pretty simple, so I won't go through that. And then also knowing induced pluripotent stem cells and how they differ from embryonic stem cells. So as I mentioned before, as we go through life, your telomeres of your chromosomes kind of shorten. So when you're an adult, your chromosomes are shorter and can have more methylation, which means it might not exactly replicate an embryonic stem cell. And again, just read this for CRISPR, be familiar with that. And then now karyotyping. So you can see here that we have different karyotypes. So you should know that we have 23 pairs of chromosomes, 22 pairs of autosomes, and then you have your sex chromosomes there. So there's different abnormalities. So there's numerical, so like abnormalities, so trisomy, monosomy. And then this is the notation you can express it in. So the total number of chromosomes, and then you specify the sex chromosomes. And then you can also specify any abnormalities after that, like this. And then you should know about your pedigrees and inheritance as well. So know that there's 2 main types, autosomal sex-linked, further divided into recessive and dominant. And then this is kind of a cheat sheet on how you can kind of determine between all the 4 types, 5 if you include Y-linked. I won't go through that because you can read through that in your own time. And this is straight from one of your tutorials, knowing the different types of structural abnormalities of chromosomes. And then basically seeing how they may or may not impact phenotype as well. So if all the material's there, it may not actually have that big of a deal on how it's expressed. And then we have translocation as well. So if it's reciprocal, it's okay, all the chromosomes are preserved. But in your Robertsonian, you have this mini micro DNA chromosome thing here that kind of just gets degraded because it's so little. It may or may not have impacts on DNA expression. Okay, question. Which of the following best describes the difference between mutations and single nucleotide polymorphisms? So I haven't gone through this, so let's see if anyone knows. Shout it out. B, is that what I heard? Or D? So D is the correct answer for that one there. So mutations occur in less than 1% of the population, whereas your SNPs need to be found in more than 1%. So that's just something you have to remember for that one there. Okay, this one here is maths. So I'll give you a second to try to work this out. So in a goat population, dominant allele A codes for black coat, recessive allele codes for white coat. They have 35 black goats, 15 white goats, which the following is true if in the entire population, 20 goats have at least 1 recessive allele. Okay, I'll just quickly go through this one because we know that 20 goats have at least 1 recessive allele. That means these 15 goats have to have the recessive allele, and 5 of the 35 black goats need to have the recessive allele as well. So if we think about the total number of recessive alleles, we have 15 times 2 because each of them would have 2 recessive alleles, so 30, plus the 5 recessive alleles from the black goats, which would just be 1 out of their 2 alleles, giving you a total of 35 out of 100 total alleles. So the answer for that one, so your recessive will be 0.35, leaving your dominant allele to be 0.65 in terms of how prevalent it is. Okay, and in order to change a child's karyotype, all the following biological materials can be used as a source except, second table over there, answer for this one? Which one? B? Yeah, so B is the only one that does not have a nucleus, so it won't have any genetic material for you to use. Easy. Okay, any questions about genes? No, we're good? Okay, easy. So cellular energetics. We have more questions, lovely. So what combination is correctly matched in terms of, catabolic, anabolic, exogonic, endogonic? Table at the front, down there. You want to shout an answer? I heard C. I don't know where that came from, but that's correct. So your anabolic reaction absorbs heat and it's non-spontaneous. It needs something to kind of trigger it. Okay, next one there. Which statements about chemical reactions is correct? Anyone? I heard D. D is the right answer. So reactions endogonic if the products contain more energy than the reactants. So I have a diagram later which kind of shows that one there. I won't go through some questions maybe. You can have a look at these questions in your own time about what they do. Okay, this one's probably something like exam type question you might see. So I will have a look at this one, see if you can work it out. Someone wants to shout out the answer. Yeah, B. So this one here has to do with temperature and how enzymes function. So you should know that kind of like the higher the temperature, the faster the reaction will take place, up to a certain extent. So there reaches a point where the enzymes then become denatured. Okay, and then have a look at this one here as well. So this one has to do with pH. Okay, so hopefully you can see here that the answer for this one is D. So we should know that in the stomach it's quite acidic. So pH of 6 and pH of 8 is going to be too high in order for any enzyme in the stomach to work. So neither will clear. Okay, so again, this is not really that important, just for kind of you to read, but you should know here the important bit is that exogonic releases energy, endogonic absorbs energy. So exo, think out, so energy goes out. Endo, think in, energy goes in. And then you should just kind of know here about delta G, so free energy. So this pretty much just tells you whether reactions endogonic or exogonic. Okay, so we can see your types of reactions here. I might look at this next one just so you can see it better. So this is quite good, these diagrams here. So you can see in exogonic, your reactants have more energy than your products because energy is going out. Even so, you still have to get over this activation energy bump in order to start the process. And then endogonic's the opposite, where your reactants have less energy than your products. And you can see here, you have a negative delta G, while in your exogonic, you have positive delta G there. And you can see the function of the enzymes pretty much lowers the activation energy for both of these reactions here. You can see this here as well, the rate of reaction without enzyme versus with enzyme. You can see it's quite quick, but then it reaches a plateau as either all the enzymes become saturated or all the products have already been made. This one here, not as important. You can just read through that in your own time. Inhibition. This one's really important. Okay, listen up. So there's 4 types of inhibition. So you have non-competitive, which you can see here. There's something else that binds to a place that's not the active site, which kind of changes the shape of the enzyme. So you call this one here the allosteric site. You have competitive inhibition, which you can see here, the inhibitor and the substrate look exactly the same. So they're fighting for the same spot here. And end product inhibition is where whatever you make becomes that non-competitive inhibitor. You also have uncompetitive inhibitor, which is when the inhibitor binds to the complex while it's already formed, preventing the product from being released. You can see that there, what that means with uncompetitive inhibitor. So as I said, basically effective temperature for most enzymes in the body, peak is going to be 37, kind of decrease after that as it gets denatured. pH depends on whatever enzyme you're looking at. Changes depending on the enzyme, and it pretty much forms a bell-shaped curve on either side of it. Okay, any questions about that?
What's the difference between non-competitive inhibition and allosteric inhibition?
They're exactly the same thing. Different words, same thing. So the non-competitive inhibitor binds to the allosteric site, therefore it's allosteric inhibition as well. Any other questions? No? Okay, easy. Next, metabolism, all you guys' favorite part of biochem. Okay, as I said, we want to focus on the big picture. So kind of just going to go through how each of the pathways work, but all of these slides kind of come together to form a massive diagram, and just know like what does what's the process called, and kind of what's released in that as well. So I won't go through this in immense detail, but you have this here to look over in your own time. So basically what this is the conversion of glucose to acetyl-CoA. So you can see here glycolysis goes to pyruvate, and then from there you either have a pathway that needs oxygen compared to one that does not need oxygen, and then that changes the pathway there. Also just know all these terms like glycogenesis, gluconeogenesis, and then also know the difference between glucose 6-phosphate and what it does in the liver compared to what it does in the muscles. So in the muscles, it can enter glycolysis straight away. In the liver, because it has the enzyme which can turn this into glucose, it can be released to the blood to regulate blood glucose levels. Okay, now we have fats. So as we can see here, this is kind of the process here. We start with large fatty acids, get emulsified to kind of microns. In the adipose tissue, we have then your triglycerides. And then just have a look at this in your own time about what happens here. And basically, the whole process to go to acetyl-CoA undergoes beta oxidation. So what you need to know here is that every 2 carbons lost, 1 acetyl-CoA is formed. So beta oxidation needs to occur 7 times in a 16-chain fatty acid. And therefore, you get 8 acetyl-CoA. So there's always 1 more acetyl-CoA you get than the number of times beta oxidation occurs. And then lastly, we have amino acids turning into acetyl-CoA. So with the amino acids, there's 2 paths that goes down. One goes to the urea cycle with the amine end. The other side, the carbon skeleton, can turn into your acetyl-CoA. Okay, and then what happens after acetyl-CoA. So have a look at this at your own time. So basically, it can turn back into fatty acids, can turn into cholesterol, can enter the citric acid cycle, or it can undergo ketogenesis. And then just the electrotransport chain, you literally don't have to know how it works. Just like you don't have to know all the intermediates of the citric acid cycle, you just need to know pretty much what is released from it. So just look at the different inputs and outputs in the arrows here. And just note this last bit here, 4 hydrogen ions turn into 1 ATP. Okay, this bit's quite important. So knowing what is happening in the fed state compared to the fasting state. So what you should know in the fed state, what it's trying to do is it's trying to turn your energy into things that it can store. So it's making glycogen, making fatty acids, making muscle, that sort of thing. So that's the premise of in the fed state, what happens. But have a look at this, it's quite high yield. And then you also have to know what happens in the fasting state. So the opposite happens. You're breaking down fat, you're breaking down muscle, all to produce glucose to fuel your body, etc. So gluconeogenesis is the important bit there. Okay, so this bit here is, remember you did like that great metabolic race, someone starts running, what energy source are they using? This is pretty much all you need to know. Start with fat, immediately you have a spike in carbohydrate use, and then this one gradually decreases, your use of fats gradually increases. All you have to know, about halfway, that's like 30 minutes in, that's when it's kind of at equilibrium, and then it swaps over from there. Okay. Read this in your own time, different lipoproteins, quite high yield, what all of them do. Again, you should know their composition, pretty much how dense they are, and what their function is and where they come from. Again, just reference slides. Okay, question, what is the difference between mobilization of stored glucose in the muscle compared to the liver? C, yes, good. So as I said there, the liver has this enzyme, glucose 6-phosphatase, which can allow glucose to be formed from glucose 6-phosphate. Next one there, what carrier molecule are fatty acids bound to when traveling through the bloodstream after being mobilized from tags? Okay, answer this one's B, albumin. So again, you'll know that once you read through those slides before. And then you should also know which lipoproteins take cholesterol away from the tissues. So you should know that this is like the good cholesterol that they kind of talk about. So which is HDL, so high density lipoproteins. So they take away the cholesterol from the tissue, which is better for you. This one is a very high yield question. It comes up quite a lot. So what is a rate limiting step of fatty acid production? Yes, A. I love that. Everyone knows that there. So that's something that gets tested a lot. Malonyl CoA A, limiting step. Easy. And then this is your metabolic race type question. So before he starts running, what's the main energy source? See, I think I heard lipids, good. So that's like your resting energy source, what's used. Okay, now he just starts 5 seconds in. What's John's primary energy source? A, good. So that wasn't on the graph, but that's something you have to know, that's the immediate source of energy. Okay, any questions about metabolism? Nope, we're good? Okay. Okay, moving on to alcohol metabolism. So once again, big picture, that's what you need to know. This is pretty much everything you need to know about alcohol metabolism. So we have ethanol, which by this enzyme here, alcohol dehydrogenase gets turned to acetaldehyde. That then gets turned into acetate. And note here that we have NAD+, which is being used. So at this stage, NAD+ is at very low levels. So you have acetate, which can turn into acetyl-CoA. But note this cannot enter the citric acid cycle because there's you need NAD+, which has already been depleted. So from here, in order to replenish the NAD+, that's all that drives the rest of it. So that's all you need to remember. You need to replenish NAD+. So in order to do that, you can make triglycerides. In order to do that, you can make pyruvate and can make lactate. So you can see here, that's pretty much what's preferred in order to replace those NAD+. So you cannot break down triglycerides. You cannot form glucose from pyruvate. So as you can see here, it's not that great for your body. So you can see triglycerides kind of builds up in your liver, which can lead to fatty liver, can lead to cirrhosis. You don't want that, I promise you. So just remember, alcohol metabolism uses a lot of NAD+, need to replenish NAD+, makes triglycerides, makes lactate. You can read the rest of the details in your own time. Okay. Okay, yapped on about this one enough, so I think you should all know the answer for this one is B. Okay, so next we have this defect in enzyme aldehyde dehydrogenase gene, and as such, there's a buildup of what molecule in the body? I think I heard B. I might have just been gaslighting myself, but B is the answer for this one here. So acetaldehyde. So that's the second step after ethanol. And next one there, Chris decides to go out and drink lots of alcohol one night. Which of these processes is occurring as Chris's body begins to break down the alcohol? Yep, so hopefully my little summary is enough. You should know this one's A. Produce lactate to regenerate the NAD+. And next one there, which of the following substances can be used to treat complications that can arise in the future from the overconsumption of alcohol? So this is kind of just something you have to remember. Okay, B for this one here, thiamine. So vitamin B derivative. So just got to know that one. Not much to say about that one there. Okay, so with these questions here, I urge you to kind of go back through them and go through the wrong answers. Figure out why the wrong answers are wrong, and think about what molecules they are, what they're referring to. So in that way, you're kind of building up your knowledge through like application as well. Okay, so that's the end of alcohol metabolism. Does anyone have any questions? There's just a question in the Zoom chat from before, the RNA, DNA polymerase stuff. With like DNA polymerase 3, we know what the function of that one is. But for DNA polymerase 1 and 2, it's not that important to know the function. With a lot of biochem, as I said before, the specific details are not that important for a lot of it. If you understand the overall picture, you're going to understand biochem a lot better and get majority of your questions right. So don't get too caught up on the tiny like details and functions of things that are not that relevant to like overall picture things. Any other questions? Okay, so let's move on then to cancer. So this is the last segment, so let's see how we go. So important to know the different types of cancer. So we have carcinomas. So those ones arise from skin and epithelial tissue. You have sarcomas, arise from bone, muscle, and blood. And then lymphomas, as the name suggests, arise from lymphoid cells. So also know leukemias arise from like white blood cells as well. So important you know these definitions here. Okay, question. What type of mutations do not cause cancer? I think I hear silent mutations, yes. So obviously the rest of them are going to have some impact on causing cancer. So increased cell growth, if that gets out of control, can cause cancer. If you resist apoptosis, so remember apoptosis is that process of breaking down cells. So if you're not able to break down cells that are damaged or degraded, then it's going to keep proliferating. Tissue invasiveness, so that enables spread. Stimulate angiogenesis. So angiogenesis is the formation of new blood vessels. So these blood vessels kind of supply the cancer, which allows it to keep growing. And E, escape immune surveillance. Obviously, it's just not able to be degraded. So silent mutations are the ones where like the base is changed, but it doesn't affect the protein that's coded. What is not a characteristic of cancer cells? A? Yes, good. So you should know that we have a larger nucleus. And then, therefore, this makes these 4 quite important to know for the characteristics. So 1, larger nucleus. B, there's changes to cytoskeleton. C, because the nucleus is larger, there's less cytoplasm. And therefore, you also lose specialized features of the cell. Okay, what's the difference between benign and malignant cancer? Yeah, C for that one, good. So benign remains localized, while your malignant invades surrounding tissue. So the rest of them is pretty much the opposite. So benign resemble your cell of origin, while malignant don't. Benign are usually okay, maybe, while malignant are quite dangerous. And then benign can become malignant. So you're just going to be careful with those ones there. So C. Okay, so it's important that you know how cancer can spread. So locally, it can spread between like walls and basement membranes, but it can also metastasize. So that's via the lymph system, via the blood system, and form secondary cancers in other places in the body. And there's different requirements of cancerous cells. So first, high-level genetic change, ability to adhere and move, angiogenesis to develop new microcirculation, and specific mechanical factors also regulate cancer cell deposition. And this is the last slide that I have. So we are looking at what are oncogenes. So this is quite important, like these definitions for you to know. So oncogenes, what are they? They're altered forms of normal cellular genes. They promote growth, while proto-oncogenes are like normal growth, development, and differentiation genes. And if we think like how oncogenes are formed, so activating mutation in DNA coding sequences can form oncogenes. Gene amplification, chromosomal translocation, those can all form oncogenes. Now, important for oncogenes here is that it only requires mutation of one of the cell's alleles. So if one of the cell's alleles is like messed up, it can form these oncogenes already. Now, these next 2 are important. The difference between tumor suppressor genes and mismatch repair genes. So the name should give you kind of an indication as to what they do. So tumor suppressor genes, they are regulatory inhibitors of cell cycle. They allow apoptosis to occur. They control different checkpoints in the cell cycle. And for this one here, it's really important that the loss of function for this one here is required in both copies of the gene in the cell. And like if you have one mutation, it's only a predisposition. While your oncogenes, if those ones are formed there, you only need one copy of that gene in order to create cancer, wreak havoc on the cell. Mismatch repair genes, they maintain genomic stability. We talked about what mismatch repair was before, and also loss of function is required in both copies of the gene in the cell. And lastly, if you just think about like what are the biological effects of cancer, so it displaces and deteriorates normal tissue, which can lead to like failure of organs and tissues, blocks vital passages of like the bowel, blood work, lymph, can cause like weight loss and fatigue, and just not a good thing to have. Okay, so that's pretty much the end of biochem for today. Does anyone have any questions about anything that's been covered? No? You guys understand everything there is to know about biochem? Oh, behind computer there.
What's thiamine's role in the cell?
I don't know. You don't need to know either. Just know that thiamine is needed. It can be used for like alcohol poisoning. Don't need to know the details. I don't know the details.
Could you explain the role of primers in DNA replication and transcription?
So for transcription, there's not really a role of primers. So primers are more associated with DNA replication. So with your transcription, I messed this up. Oops. Okay, so as you can see with transcription here, there's not really a role of like primer. So you don't really need the primer. What you have here is like a promoter region and an initiation site. That there is where the RNA polymerase 3 can directly bind to and just start elongating the mRNA. So where you find the primer will be on here. So you have RNA primers for DNA replication. So RNA primers are placed there by DNA primase, but ultimately those are removed and replaced with DNA bases after the strands have been formed.
Does the RNA primer refer to the fact it's used in RNA synthesis as well, or just because it's made of RNA?
It's just the fact that it's made with RNA. So that's why it has to be removed, because it's not actually a DNA base, it's an RNA base. Anything else? Okay, cool. So that brings us to the end of biochem. If you do have any more questions, do you have a question? No. If we do have any more questions, feel free to message me on Messenger. I will attempt to give you a good explanation. I'll hand back over to Renee.
Well done. That was hella quick. So we're running like an hour ahead of schedule, which is really, really good because he's usually run behind. So we're going to get straight into it with micro. Yeah. A little skip. Yeah, I don't know. Do we want to do micro? But yeah, there we go. All right. And we've got plenty of time for questions, so keep them coming in. Put your hand up if you want to ask any. Even during the presentation, if you've got questions, just put your hand up and we should be able to. Oh, yeah. I don't want the mic. Okay, come. Okay, we can do that now. Yeah, okay. Izzy? Do you want to do more biotech stuff? Yeah. Yeah, I should ask them. Yeah. It doesn't, like, if you don't want to. All right, we're going to do a brief overview of biotech. So if you could just pay attention for a little bit longer, and then we'll give you a 5-minute break. get into it. Oh, the network keeps turning off. I want to guess
Oh, are you on guess what?
because I've signed into a different account because I don't want like stuff to pop up during the.
Okay, we have a request to look at biotech in a bit more detail because I just said to read it yourself. So I'll go over how a lot of this works. So just settle down and we'll get started with this soon. So it'll be quite brief, just kind of go over these different biotech processes. If everyone wants to find a seat quickly. Yeah, if everyone can find a seat quickly, that'll be much appreciated. Just do 5-10 minutes on this and then we'll have a break, short break. Okay, let's get into this quickly. So first, farming. So what this means is that you have this gene here that you want to replicate. You want to make a lot of it. So you insert this into a nucleus, as you can see like this. It's an animal embryo. And then this is transplanted into a animal. So this offspring now will have the desired protein being made. So this protein is then extracted from the offspring, which has pretty much this chromosome here. So an example of this, transgenic goats secrete antithrombin 3, which can use to prevent blood clotting during surgery, which is present in their milk. And then for therapeutic cloning, it's important to know the different types of stem cells. So totipotent, they're not generally used because these ones, like it's very early on. So these can give rise to any cell in the human body. And it's also present extra embryonic tissue, like placenta as well. So a fertilized egg is considered like pretty much the only totipotent cell. Pluripotent is the one that's quite usable. So they can give rise to any cell in the body, but not placental ones. So these are what you kind of refer to when you say embryonic stem cells. And then multipotent is unusable under certain circumstances because they only give rise to cells in their own lineage. And then for gene editing here, so it requires modification of existing cells and tissues in the patient. And then a vector is kind of used to deliver the new genetic information to the patient. But just know now that it's still very early on in its technology. So a lot of things can go wrong, not really that widely used yet. I'll go into CRISPR-Cas in the next diagram where I have that there. Okay, so for recombinant DNA, if I just go through the process here quickly. So first you need to isolate the gene of interest. So that's used with endonucleases, which are restriction enzymes. So they cut DNA at restriction digestion sites. From there, you can see here that it's the gene that whatever cut the gene cuts the vector with the same restriction enzyme, so it's compatible. It has sticky ends, so it can kind of attach to each other. So this vector is usually a plasmid. So remember, plasmids come from prokaryotic cells. So we have sticky ends here, as you can see here. So it's kind of overhanging on one side and then overhanging on the opposite side there. So as you can see now, we have a plasmid with this DNA of interest that we want here. So they just use ligase to bind it together to form recombinant DNA. Then it's inserted into the host cell. It's inserted into a vector via heat, electronic currents, which is normally a bacteria cell as well. It's allowed to replicate, it's cultured successfully, and before it gets cultured, it is usually exposed to something like an antibiotic, so something that can select which bacteria actually have the gene that you want. So you're only left with cultures that have the gene of interest. And then you pretty much just extract the protein once it's made, and then you just have a bunch of these proteins which is coming from the gene of interest. And then, as I mentioned before, there's differences as to why certain host cells are used. And then polymerase chain reaction, as I said, first step, denaturation. So it's heated, pretty much just separating the two strands. Annealing, so this is when the temperature is lowered. You put it in a solution with like these small primers, which allow these DNA primers to attach. And then finally extension, so it's heated up again. And then it's added with Taq polymerase, which is a polymerase in bacteria, I'm pretty sure, which is 72 degrees is its like optimal temperature. So from there, it pretty much just extends the DNA. So now you have 2 strands of DNA. So every time you do this, you pretty much double the amount of DNA. So it grows quite exponentially. And then somatic cell nuclear transfer. So this is quite important. So you have 2 things. So you have a egg cell and you have a somatic cell. The egg cell is the one where the nucleus is removed and it's replaced with the nucleus from the somatic cell. So this is kind of the genes that you want to replicate. So now you've kind of made an artificial embryo. So you have an egg with the nucleus of the genes that you want. So this one is a clone of this somatic cell. Then when it duplicates, it becomes an embryo and it becomes a clone of this cell here, and it can form into different cell types. So note here that it's approved for research, but embryo development beyond 14 days is not permitted. You can't implant it into a woman or animal's body, and you can't produce embryos by egg and sperm for research. And then I talked a bit about this, but basically they just induced pluripotent stem cells are just reprogrammed by targeting 4 specific genes. And then these can then be stimulated to produce various cell types, as you can see here. So basically the point of this is that this adult cell comes from the person you want to treat. So as such, all the DNA is the same, so it's less likely to get rejected by the body. And then we have CRISPR-Cas. So CRISPR-Cas, so like CRISPR sequences are things that are seen in like bacteria, and they're kind of separated by these like repeats here. So the spacer. So these spacers are remnants of prior virus invaders. So when a virus with a target unique sequence invades a cell, an RNA is made that includes a CRISPR-Cas sequence, as we can see here. And then another RNA is added, as you can see the green one here. And then we have this Cas9 nuclease, which then binds to the mRNA. So what this does is basically it binds to the unique target on the invading viral genome. The nuclease cuts the viral DNA, which inactivates it. So as you can see, it's cut. So this prevents viral reproduction in the bacterial cell. So an RNA can be made in the lab that has any desired target sequence and the Cas9 binding sequence. So it's basically like inactivating the gene. So once this is done, you can kind of assist repair as well. So you can put in a repair sequence, but what this basically does is it takes out like, this gene, it becomes inactivated. So if it's faulty or if it's a mutation, you won't see the effects of it. And then, so that's pretty much where CRISPR-Cas comes from, stems from bacterial viral origins, but we can see how we can use that for DNA therapeutics as well. Cool, so that's biotech explained in more detail. So we'll finish up by Ken for today. Yeah, cool. Any questions? Lovely. Thank you.
If you all don't get 100% in biochem, we've got problems because that was literally everything you need to know. We're going to get into, we'll do a 10-minute break first because I think everyone has conversations they need to have. So come back at 11:20 to get into micro. Get excited, get keen.
What do you mean that's for micro?
I reckon you can do it. I reckon. We get like a Facebook confirmation of whose birthdays it is, like ask around. We can do it. That's
I want to give you a good reply, but everybody in this room is birthday.
right, but
Testing.
Ah,
How good?
pretty good. That's pretty good. Yeah.
Testing. Okay, nice. Unless everyone.
All right, we're gonna get started. Couple more minutes to let everyone come back in. It's just how I sound like usually.
No, laser pointer doesn't work.
Is what? Oh, I think it's.
The laser pointer doesn't work.
Oh, is it because it's too far away? Yeah. Fuck.
It's all good.
Damn, that would have been sick if we could just get the laser pointer to work.
Sit at that.
All right, is everyone here? Calm. Okay. Well, we've got Micro now by the Micro King himself, Alex. So I'll let him take him away now.
Cool. I'll get started right away just because I don't want to run overtime. There's my email if you want me to shoot me an email, or you can message me if you have any questions, and ignore the PowerPoint note there. So just some references. You can have a look at that in your own time. Thanks to Meredith and then the past swap back revision lectures. And if you haven't had a look at the exam breakdown, I recommend you do. Micro is only really applicable for the second exam, and it's 20 marks. So it's the majority of paper 2, but not the majority of all the marks, but still worth to know. Nice. Okay, so today we'll be going over 4 different parts. We'll go over microbes, diagnosis and control, and innate immunity. And if we have time, we'll go over the questions. If not, you can go through those in your own time. So just some revision before we get started. I'm sure you've gone over this already, so I won't go over this too in depth. Prokaryotes and eukaryotes, the main difference is prokaryotes do not have membrane enclosed organelles, and they have a nucleoid, so they don't have a nuclear envelope. Contrast this with the eukaryotes. You can see they've got, mitochondria, Golgi apparatus, endoplasmic reticulum, all that good stuff. So yeah, and if you do want to remember all the differences, you can memorize it, or you can use a mnemonic. DORA is 1. You can go through that yourself. But it's DNA organelles, reproduction, average size. And in terms of what the prokaryotes include, they include bacteria and archaea, and eukaryotes include animal, plant, fungi, and protists. And another concept that doesn't really fit anywhere else, so I'll put in the revision, is symbiosis. So that's just any long-term relationship between two biological organisms. So they include mutualism, commensalism, parasitism. Mutualism, it benefits both people. Commensalism benefits one species but doesn't harm the other. Parasitism harms one, benefits the other. You may have also learned like competition and predation in, I don't know, science, but they're technically not symbiosis because it's not a long-term relationship. It's just an ecological relationship. All right, microbes. So some quick facts. If we did this on PowerPoint, that would spin around and stuff, like 3D graphics, but that's all right. So they're unicellular, they reproduce via binary fission, or very standard of prokaryotes. They also have linear circular DNA and 70S ribosomes. And how that comes into play, later on we'll explain. 70S, because they're different to the eukaryotic ribosomes, you can target them using antibiotics. Distinguishing bacteria, there are a couple ways you can distinguish specific bacteria: shape and specialized structures, response to gram stains, toxins, oxygen requirements, and nutritional requirements. Now, the ones with the asterisks there, they're not explicitly mentioned in your lectures, but I figured might as well include them. All right, so we'll go over shapes first. Main three are cocci, bacilli, spiral. Pretty self-explanatory. There are more different shapes, but those are the main ones you have to know. You can see that Enterococcus is diplococci. E. coli is bacilli, and then you got the spiral one. I'm not pronouncing that. But each of those families, you can subclassify them depending, example, how many spheres they have, how many rods they have. So you've got coccus, diplococci, bacilli, diplobacilli, and yeah, sure you can figure it out. Next is specialized structures. So this is just a table summary going over the structures. You would have learned capsule or glycocalyx. Technically, they're two separate things, but if you wanted to learn the capsule and slime layer together, you can also do that. So the main distinguishing thing is a capsule is more tightly associated, so it can't be washed off, whereas a slime layer can be washed off, very loosely associated. But the main functions is protection from phagocytosis, compounds, prevents dehydration, and the rest over there. You've also got flagella, so that helps them swim. Pili, also known as fimbriae or adhesion molecules, they help bacteria adhere to surfaces and adhere to each other so they can form biofilms. And then endospores. Endospores aren't really structures of bacteria, but they're things that they can form, and they're dormant structures which are formed during unfavorable circumstances. And that just protects them and allows them to reactivate when circumstances are more favorable. All right, so these are just images of what we've gone over. You can see the top left image. Laser pointer doesn't work, unfortunately. 1 is the capsule, 2 is the slime layer, 3 is a biofilm. You can see the flagella underneath that, endospore formation in the middle. And on the right, you got the pili, adhesion proteins, and you can see attaching to the surfaces on that electron microscopy slide. And then next is response to gram stain. You got gram positive, gram negative. One stains purple, one stains pink, and that's due to the structure of the cell wall. So gram negative is purple, and that's because it has a really thick peptidoglycan layer. It can trap the dye, whereas gram negative, it has a thin peptidoglycan layer, and it also has an outer membrane. So the purple dye is washed out, and then the counter dye can stain it, and that's the pink. And notable example, probably should remember, Staphylococcus aureus, gram-positive cocci. And then you've also got gram-neutral, gram-zero, so these don't stain. The only one you really need to know for This course is Mycobacterium tuberculosis. So because of the mycolic acid, none of the stains actually stay. So we use an acid fast stain instead, and that stains red. All right, next we'll move on to toxins. There's exotoxins, endotoxins. Once again, not included in the lectures, but if you want to know, you can classify bacteria or see what it is based on the toxins it secretes. You got exotoxins, it secretes it out. Endotoxins are part of the lipopolysaccharide. It's actually synonymous now, but yeah. Oxygen requirements, you got the 5 listed there. They're pretty self-explanatory, so I won't go too in-depth into them. Obligate aerobes has to have air, oxygen. Obligate anaerobes has to have whatever. Now the numbers on there and the images do match, and you can kind of see like obligate aerobes, they need air, so they go to the surface. Obligate anaerobes, they don't need air, so they go to the bottom. I'm sure you guys can figure that out. And nutritional requirements, also pretty self-explanatory, and we'll revisit that when we do the diagnosis. So using agar plates and all that jazz. And this is just, well, the next couple slides will just be like tables and summary diagrams of notable bacteria. Nice. Hello. Sick piano. Now, you don't really have to know all of these, like memorize it. I wouldn't say it's super high yield. You will get to know them eventually as you do more lectures, experience more, learn more. But do try and remember the main ones like Streptococcus aureus. I'm being upstage right now, damn.
Do you want to ask a question?
I will have time for questions after each section, but
You just like talking to yourself, that's okay as well. Alright, calm. He's looking up a question on his phone, I reckon.
just being aware of the time, I reckon we'll try and send in the chat if you have a question, and I'll get back to it. And I will revisit it after this section. Like,
Someone just text him to mute himself and turn off his camera, please.
Alrighty. Okay, never mind. There we go. All right. So the main ones really Streptococcus aureus, Streptococcus pyogenes. Actually, they're all pretty important, but if you have to remember one, Streptococcus aureus, Staphylococcus aureus. Remember that it's the grape-shaped ones with the lots of purple ones. Yeah, great, self-explanatory. And then this is just a flow chart of gram-positive cocci, gram-negative cocci, gram-positive bacilli, and gram-negative bacilli. And you can go over that in your own time. It's not super high yield, but worth knowing. All right, so that's the end of bacteria. Are there any questions from here at the moment? People present? And I think chat's okay. All right, so we'll just move straight on to viruses. Once again, would have been a nice graphic. So viruses are non-cellular, so they're often termed obligate intracellular parasites because they require being in host cells in order to proliferate, and they're detrimental to us, hence they're parasites. In terms of distinguishing virus, same thing as bacteria, you can use the type of nucleic acid, the capsid morphology, the presence or absence of an envelope, host, so with what infects, and the disease it produces. For example, like influenza, like flu. We'll go over each of these. So in terms of its structure, the genome can be RNA, DNA, or it can be single-stranded, double-stranded. Pretty self-explanatory. It can be a mixture of the both. And the genetic material can also be a single molecule or multiple molecules. For example, influenza viruses have 6 to 8 segments. Then you've got the protein capsid, which surrounds the nucleic material. And you can have different shapes. The two ones you need to remember really are helical, also called filamentous, and polyhedral. It's usually icosahedral, so there's 20. And that's also called a spherical one because it approximates a sphere. You also have like other n equals whatever, and that's why there's polyhedral and spherical separate in the image. And you also have complex ones like bacteriophage, but not super necessary to know. And you've also got a lipid bilayer in some viruses which surround the nucleocapsid, and they also contain glycoprotein spikes, which are the spiky bits. Yeah, and they use that to enter the host cell. All right, in terms of its replication, at this point in time, you only need to know the broad steps, like these 6 steps. Attachment, so the virus attaches to the host cell. Entry, so through those glycoprotein spikes we talked about, or through another molecule on their capsid, they get bound and then endocytosed. The capsid is then uncoated, and then the viral components are synthesized, transcription and translation. Translation for the proteins, transcription for the genetic material. Assembly, so it assembles all the components into the virus and then it's released either by budding or cell lysis. So cell lysis will result in the non-enveloped viruses because, yeah, and then budding, because it buds off from the actual cell, it will form enveloped viruses. Now, next semester, you will learn the different classes of viruses. They're based on the genetic material inside them, and that affects how they replicate. But the general gist is no matter what type of genetic material is in that nucleocapsid, they eventually want to get to mRNA because from there you can go pretty much anywhere. And then clinically relevant viruses, they're all pretty relevant. Once again, you don't have to rote memorize every single one. You will remember them as you go through it. Remember, herpes viridae never goes away. That's a good one. Influenza, yeah. Influenza can undergo antigenic drift and shift. Yeah. All right, any questions on viruses? Mm-hmm. But yes.
If the capsule is made up of lipopolysaccharides, how can
You want to repeat it on your mic? Or do you want-
Yeah, do you want to repeat it for everyone?
If the capsule is made up of lipopolysaccharides, how can gram-positive bacteria have a capsule?
Yep, so we'll just go back to that slide. It might take a while. So the lipopolysaccharide is part of the cell membrane. So bacteria have cell membranes, and the capsule/slime layer is like an extra tidbit outside the cell membrane. So it's like a separate thing. It's like if you have a cell and then the capsule, think of it like spray paint. You just coat it over the cell membrane. So the LPS layer is actually part of the cell membrane, like the peptidoglycan and the lipopolysaccharide. And then outside of that, like beyond that, is the slime layer and the capsule. Does that make sense?
So the capsule is not made up of
I believe it is a completely different thing. I'm not sure what exactly it is made of. I've forgotten that. Past me remembered, but I can get back to you. Do you know what the LPS is made of? I mean, sorry, the capsule, glycocalyx. Yeah, I can get back to you though. Yeah.
Have we got any other questions?
Easy. All right, next, fungi. I like to think.
Is that a question over there or just I can't?
Just eating. It's all right, fungi. All right, fungi, they're eukaryotic and they're obligate aerobes, so they need oxygen. In terms of structure, they have chitin, beta-glucans, and mannose in their cell membrane. You can see in the right, so slightly different from all the others. And the cell membrane contains ergosterol instead of cholesterol. Same function, it's just different, quirky like that. And they can undergo both sexual and asexual reproduction. This one, you can, just have a look over it. In terms of classification, you have molds, yeast, dimorphic. That's the ones relevant for us. You've also got mushrooms, pretty yummy or not, but we don't cover those, unfortunately. So mold is the first one. They're basically fungi which extend by growing hyphae, which are long branching structures. You can kind of see the branching over there, and the molds is on the right. The fuzzy bits on the end of the hyphae are called sporangia. They're spores and how they, spore and reproduce. And then they form something called mycelium. So a bunch of mold forms in the bottom right-hand corner, a network, and their mycelium. In terms of the hyphae, you can also have septate hyphae and non-septate hyphae. And you can see the main difference is whether or not it's segmented, like separated, or one long tube. In terms of yeasts, they grow via budding instead of extending the hyphae, and that's a form of asymmetrical reproduction. So this is different from binary fission. You can see it's asymmetric in the top left image, the left image. And they can also form something that looks like mycelium called by forming pseudohyphae. And they're still yeast cells, still discrete cells, but they just look like it because, yeah. And then dimorphic is a little bit of both. Can exist as either, usually in response to temperature. You can see usually either dimorphic or yeasts are the problematic ones because they're easily transmitted in terms of like pathogenesis because they're good at body temperature, 37 degrees, like they're in that form. All right, so in terms of how fungi can actually cause disease, there's a couple of different ways. You can have allergies, you can have reactions to the actual toxin itself, the mycotoxin, or you can have mycoses, which are the actual infections due to the fungi. And the mycoses, we'll go more in depth, is broadly classified into cutaneous, subcutaneous, and invasive. And the brackets are just alternate names. So cutaneous ones, they're usually transmitted via contact. And the example we're given in our lectures are dermatophytes, and they grow wherever there's keratin. So your skin, hairs, nails. You probably don't have to remember these, but those were the examples given. Trichophyton, rubrum, and epidermophyton flocosum. But they do cause tinea, which is also known as ringworm, because it forms those ring-like lesions on your skin. And the tinea, and the second word in that is usually referring to where it is. So tinea capitis in the hair and scalp, tinea pedis is athlete's foot, so on the foot, and tinea is the nails. And risk factors for this is warmer climate, just because they like the warmer areas, sweaty creases, and poor hygiene. Hey, what do you have against sweaty creases? All right, next, subcutaneous. You got inoculation, so that's primarily how it's transmitted. The example we're given is Sporothrix schenki. It causes rose gardener's disease, called that because it's usually transmitted through the thorns of roses that have been left in the dirt, so rose gardeners get it. And it may form a single plaque or multiple plaques that erupt along the line of lymphatics. So that's probably following their lymphatic line. And finally, we've got invasive, usually spread via inhalation. That's why they have their masks. And the examples we're given is Candida, Cryptococcus, Pneumocystis, and Aspergillus. We'll go over each of these More specifically, you don't have to remember every single detail, but whatever you retain, since it's technically accessible, can't hurt. Candida, dimorphic, main one is Candida albicans. It's opportunistic and is found within the normal flora. And the main manifestations it has is either superficial infections or it can cause thrush, especially after antibiotics. It can also cause esophageal candidiasis in immunosuppressed people, and it can even go into the bloodstream, so candidemia in immunosuppressed and IV drug users. And that can also lead to infective endocarditis, which is infection of the heart, basically, which we'll cover later. Cryptococcus, it's a yeast. C. gattii and C. neoformans are the ones examples given. C. neoformans transmitted via bird feces, C. gattiis, river red gums. Usually it's a respiratory one, causes pneumonia, can also cause meningitis if it gets into your central nervous system, and it can also infect your skins and bones, causing painful lesions. And you can see like x-rays and lesions on the right. Pneumocystis is yeast-like. Honestly, I don't know what yeast-like means, probably, yeah, but P. giravechi, airborne transmission mainly affects immunocompromised people, but healthy individuals may be carriers, and manifestation is pneumonia and any associated respiratory symptoms. And then lastly, aspergillus, inhaled from the environment, and it can cause respiratory symptoms either through infection, usually through immunocompromised people, or through an allergy. So that causes something called allergic bronchopulmonary aspergillosis. And that's more common in people with cystic fibrosis and asthma. So any chronic respiratory conditions. And you can have spread through the blood, leading to infection in the brain, kidneys, and heart. Once again, probably don't need to know all of this. If you want to revise this over the slides, feel free. Just a general understanding of mycoses should be fine. Don't take my word for it though. And then medically important eukaryotes, that will also include the protists that we'll go over later. All right, any questions on that? Nope. Are we expected to remember details of specific viruses for the exam or just general info about viruses? So for your first semester exams, it's mostly just knowing the general mechanism. Obviously, since it's in the lecture, it's technically accessible, but the learning objectives don't expect you to know specific ones. You will have lectures next semester which go over more specific bacteria and viruses. And then so next semester's exams, you might be expected to know a little bit more. But in our exams last year, notably, there was a little bit of it was a lot of effort to remember them and not much marks on the exam for next semester. So yeah, do with that what you will. All right, next, another question is, what is the relative importance of the pathogen characteristics, disease cause mode? I'll answer that in chats after, just because I'm concerned on the time. All right, parasites. Parasites are also eukaryotic. You've got protozoa and helminths. You've also got ectoparasites. Once again, not relevant for our course. Ectoparasites are like lice, ticks, fleas, mites. Good protein. And then, yeah, protozoa are unicellular, helminths are multicellular. So the specific ones going over your lecture, well, two of them are malaria and trichomoniasis. With malaria, it's caused by 5 different Plasmodium species. Really, you only have to remember P. falciparum, and that is the most severe form of malaria. And it is transmitted through, or all 5 of these are transferred through specific species of female mosquitoes. and it can also be transplacental. And it affects hepatocytes and erythrocytes, so red blood cells and liver cells. And notable information is that for malaria, prevention is key. So prophylaxis, nets, mosquito repellent, things like that. And any fever that a returning traveler has should be considered as malaria unless otherwise proven otherwise. Next, Trichomonas vaginalis. That's a sexually transmitted disease. Infects the urogenital tract. And most people are asymptomatic, but it does present more symptomatically in women. Symptoms are dysuria, dyspareunia, discharge. So that's painful peeing, painful intercourse, and discharge. Can also predispose you to other STDs. And that's a little trichomonas over there. Trichomonas. And then this is more in depth of the malaria life cycle. The learning outcomes in your lectures don't expect you to know these. They just want you to appreciate this, whatever that means. But you can see affects the red blood cells, infects the livers, goes to the mosquito, yada, yada. The next two are toxoplasmosis, so Toxoplasma gondii. It's zoonotic, comes from cat feces, but it can go through intermediate hosts. And it's a GI infection. So you have the GI symptoms, you also have flu-like symptoms. And it's usually fine for most people, but it's very severe for people who are immunocompromised and pregnant. And it can form cysts in your tissue if it spreads. And that can be also reactivated. And the Giardia, contaminated water source. Some of you may have heard this. It affects the small intestines and causes watery and greasy stool, then greasy stool. That's the notable symptom, but it also causes a host of GI problems. And once again, similar to malaria, prevention is key. So boiling water, filtering water, make sure the water you're drinking now is clean. And then once again, more in depth, you can look over that in your own time. And then helminths, they're worms. There's no consensus on how to organize them, but the two notable groups that we explored are nematodes, roundworms, and platyhelminths, flatworms. So nematodes, you've got hookworms, filariasis, human pinworms as examples gone over in your lectures. And then platyhelminths, flatworms, so you've got tapeworms and flukes. And the specific example we had in the lectures were blood flukes. If you're curious, the difference between tapeworms and flukes, you can see on the right image, the top left box and the top right box. One is like segmented, and that's the cestodes, and one is not segmented, and that's the trematode, the flukes. And then these are just the life cycles of each, slightly different for each, but yeah, basically all you're expected to do is appreciate the diversity of these life cycles. So feel free to have a look at them in your own time. One notable thing to note when you do go over these in your own time is when they can be diagnosed. So when they can they be seen? Usually it's through eggs in the feces, but sometimes it's through blood tests at certain times and whatnot. And then those are platyhelminths, flatworms, and then clinically relevant eukaryotes, same table as before. Any questions on that? Alrighty, diagnosis and control. That was the bulk of it, so now everything should be easy breezy. Diagnosis, commonly you'll hear MCS, microscopy, culture, sensitivity. Then you've also got other tests like serology, molecular techniques, and as well as other specific tests like catalase tests. So the catalase test is on the bottom right. You can see you've got hydrogen peroxide, and catalase basically breaks that down into water and oxygen. And some bacteria will have catalase. So if there's bubbles, that basically indicates there is catalase present. Oh, and a note with serology. Serology is different from blood tests because the only test for antibodies. Something to note. All right, in terms of culturing, this is mainly for bacteria because that's what we have a focus on. This is a summary of the different media you can use. I won't go through all of it, but nutrient agar is your stock standard one. Then you got ones that can be selective. So it only allows certain bacteria to grow, and that can be useful, especially if you are swabbing something that has a lot of normal flora. And then you've got something differential. So it will show you a change depending on the bacteria. For example, depending on the hemolysis, like alpha, beta, gamma, horse blood agar will turn different colors, depending on whether or not the bacteria uses lactose and breaks it down into lactic acid. You can use that with the McConkey agar. And then mannitol salt agar, a differential one is Staphylococcus aureus. It actually turns yellow. That's why sometimes they call Staph aureus golden staph. And then you can see images of these. Top left one, I'll go left to right, top to bottom. So nutrient agar, horse blood agar, chocolate agar, which is basically a heated up version of horse blood agar. Then you've got the McConkey one. And then you've got the, what's the name of it? Golden staph one, mannitol salt agar. And then this is the bacterial flowchart. Once again, you don't have to remember every single part of it. Probably just know the main ones like S. aureus, S. pneumoniae, S. pyogenes. Yeah, those are just the main ones. Although it's very unlikely that I'll have a question on this on the exam. Next is the chain of infection. So the chain of infection is important to understand because we need to break the chain. The chain is a way, or all the links that a pathogen needs to propagate and spread disease. And so knowing these, we can break the chain at any of these locations to stop the spread of the disease. So you can have a look at that in your own time. Pretty self-explanatory though. And in terms of controlling the reservoir, which is one of the links in the chains, you have cleaning, disinfection, sterilization. Cleaning is just removing the visible dirt, soil, and it's a prerequisite for disinfection and sterilization. Disinfection, removing most viable organisms, sterilization, killing all viable organisms. So that includes the endospores that we mentioned earlier. And then antiseptics are a subset of disinfectants that removes and kills microbes from living tissue. So like skin, during surgery, all that. So this is just a summary of all the ones covered. Soap and water cleaning, thermal treatments is most of these are either disinfection or sterilization, depending on the chemical used or the method used. So you can see moist heat and dry heat. They're a form of sterilization. Moist heat is much quicker because it uses pressurized water. And you can see on the right, those are the instructions, how long you do it for, what temperature. And then the only one there is boiling, which is a form of disinfection. And the threshold is basically, a rule of thumb is disinfection is below 100 degrees, sterilization is above 100 degrees. Obviously it has to be for the correct amount of time, but that's the general rule of thumb. Radiation treatment, you've got non-ionizing and ionizing. With non-ionizing, it does not penetrate well and it can damage tissues and plastics. So it's usually used just to clean like surfaces or the air or water. And ionizing can penetrate. So it can be used with packaging and treat heat-sensitive materials and some foods. And then chemicals, you don't really have to remember all of them. You can remember, like the pink bottles in your Clean Skills is chlorhexidine, iodine, alcohol, I'm sure, you know, but you can go through that in your own time. All right, now in terms of using these sterilization and disinfection techniques, when to use these depends on this Spalding classification. So you've got non-critical items, which only in contact with intact skin. low-level / intermediate level disinfection. Semi-critical touches mucous membranes and non-intact skin. High-level disinfection, and critical ones are that go instruments that go inside the tissue or inside the bloodstream, and you need sterilization for that. And just some definitions. You can go over this yourself. Asepsis, medical asepsis, surgical asepsis, fomite, nosocomial infection. All right, next is controlling the infectious agent itself. And you have antimicrobials do that. These are specific to certain types of microbes. So antibiotics are for bacteria, antivirals are for viruses, et cetera. But we'll focus today on antibiotics. So those are the major targets, the 5 targets: cell wall synthesis, protein synthesis, nucleic acid synthesis, metabolic pathways, and cell membrane function. And these may also be bactericidal, which kills the bacteria, or bacteriostatic, so it inhibits the bacteria. Doesn't kill it, but yeah. Right, antimicrobial resistance, just a definition, it's ability for a microorganism to resist the effect of the antimicrobial, basically. And so mechanisms of antibiotic resistance in bacteria, you can have genetic and non-genetic causes. So you can just have like the innate resistance of a bacteria. For example, Mycobacterium tuberculosis, as you recall, they have that thick mycolic acid layer, and that can prevent some antibiotics from binding. You can also have acquired ones, for example, alteration of target site, modification of the antibiotic itself, destruction of the antibiotic, and preventing access to the target site. And those are all summarized within the right image, basically. So you can see modification of antibiotics can be using an enzyme, destruction also enzyme, and preventing it accessing the target site can be done by the efflux pump.
I think we did a lot of this, yes.
Oh, we did? Easy.
Mitch went through it.
Easy peasy. All right. No, we can just skim through. Like, I won't go through it in depth. Yeah. Next, transformation, conjugation, transduction. That's how they get it. If you weren't there yesterday, you can go over these slides yourself. And in terms of like the genetic material, you have plasmids and transposons. Plasmids are the circular DNA, transposons are the movable DNA that can jump between the main chromosome and the plasmid. And then the implications of this, really all you have to remember is that anytime you use antibiotics contributes to AMR. Remember MindMe, you can go through that yourself. Hang on one moment. All right, so next we'll go over innate immunity. This will have some overlap with your next lectures by James, which is on immunology, but this was covered by a microbiology lecture, unfortunately. So for the bits that he will go over, I will skim over. Don't worry, he'll reintroduce those topics, and if he doesn't, feel free to ask any questions at the end or go over these slides in your own time. Like this. Yeah, so the innate immune system, you've got 3 lines of defense. The innate immune system comprises the first 2. So the first one is to prevent the actual infection. Second line is the non-specific responses to the infection. So the first line of defense, you have those barriers: anatomical, mechanical, chemical, and microbiological. Microbiological being the normal flora. You can go over each of them yourself, but it's probably useful to know all of these, really. In terms of normal flora, contains both resident and transient flora. So resident are the ones more firmly attached to your body, transient, more transient. And most are commensalistic, but some are mutualistic. Some can become parasitic if, the circumstances permit. So there's dysbiosis, there's a cut in the skin, someone's taking antibiotics. So they're termed opportunistic pathogens. And they have a couple functions. So the non-pathogenic functions, like the good functions, they inhibit colonization of other bacteria via competition and producing certain compounds like lactic acid, things like that. So that can prevent us from some infections. It also has metabolic functions such as synthesizing, excreting vitamins, and it helps train our immune system. And sterile sites to remember are any internal organs, like hopefully sterile, any internal body fluids like your blood or cerebrospinal fluid, and the lower respiratory tract and the upper respiratory tract. And for the last two, you can just remember whatever's closer to inside your body is sterile. So the upper respiratory and the lower urinary is not sterile. All right, second line of defense consists of these three things. You got immune cells, soluble proteins, and then inflammatory response and fever. For the soluble proteins, we only go over the complement system, but I've attached an image of interferons as well, because you'll learn that next semester, but you don't need to know it for this semester. So this will be going over in James's lecture as well, but regarding like specific functions of these specific cells. But you can classify the innate immune cells into two different groups. You've got phagocytic ones and degranulating ones. Phagocytic, neutrophils, macrophages, dendritic cells, monocytes. Degranulating ones, NK cells, eosinophils, mast cells, basophils. Now, some of the degranulating cells, the first two, notably, use that degranulation to kill microbes. So you can see in the bottom right image, using perforin and granzymes. And then you've also got some that only release cytokines and propagate, in the inflammatory response. In terms of the complement system, it's just a series of proteins in your blood that help out. So there are three activation pathways. You've got the classical, so that's the antibody and the complement binding to the antibody. You got the alternative, which is a complement binding directly to the pathogen, and you got the lectin pathway, which is a complement protein binding to a mannose binding lectin, which binds to the pathogen. So they all bind to the pathogen. It's just whether or not it's done directly or indirectly. And the general gist, you don't have to remember every single molecule, is that once it's initiated by those three pathways, it eventually leads to the center of C3 convertase. And then those C3 convertase leads to the three different responses, which is either inflammation, opsonization, or cell lysis. And that's through the membrane attack complex. Yeah, opsonization, good question, is basically tagging a pathogen. So imagine the complement being like a bright flashing light for the macrophages. Once you attach to the protein, macrophages, dendritic cells, they can see the complement protein, and it's been tagged for death, basically. You can see it's the rightmost one with the macrophage eating bacteria, if you can see. Yeah. Interferons, this is the image, you can go over in your own time. It's released in response to viral infections. Next is inflammatory response. This is a huge slab of text, but this is basically the inflammatory response in detail, going over all the nitty gritty for those of you who like those. I know I did. So PAM stamps, so they're pathogen associated molecular patterns, and then damage associated molecular patterns. So they're things that signal infection or damage, and that initiates all that good stuff. You can read that in your own time because it's not necessary to know it's this in depth, but in the lecture, this is how in depth I had it. So you got the splinter puncturing, that introduces bacteria, that causes injured cells to release histamine, causing vessels to dilate. And then through those dilated vessels, neutrophils, macrophages move through the vessel walls. Neutrophils are the first ones that come to the site of infection, and macrophages are derived from monocytes. So they're monocytes in the blood, and once they go into the tissue, they're macrophages. And the last one, bacteria engulfed and destroyed by phagocytosis. So you can either go that very in-depth one or somewhere in between. Yeah. And last is fever. So that's just a temporary increase in body temperature used to enhance immune cell function as well as restrict the reproduction of many pathogens. And the reason why fever occurs is when macrophages phagocytose pathogens, they can secrete things like pyrogens, subset of cytokines, and that signals the hypothalamus to raise your body temperature. All right, so these are just some questions. How you guys feeling? Do you guys want to go over it or do you guys not? Let's do it. Let's do it. Easy. All right. But before we do that, any questions on what we just covered? Lovely. All right. And courtesy of Josephine Owens, she made most of these questions in the 2019 SWOT-VAC. All right, first question. We'll go table by table because I want to spread the love. So we'll go this table first. Which is the most abundant white blood cell? Nice. Next table. Which is not a fungal infection? Ringworm, D? Ooh. So remember, ringworm is the cutaneous infection caused by dermatophytes. Aspergillus is the invasive one, and so is Candida. I have no clue what Chagas disease is. I think it's a parasitic infection. But yeah. All right, next. Which part of the fungal cell is most useful as a target for antifungals? Let's take a guess. Yeah. So they said A. The correct answer is, yeah, it's B. Nice. So the reason why we use ergosterol is because it's only found in fungi. You don't want to use something that's found in our own cells because attacking our own cells would not be fun. So remember that fungi have ergosterol instead of cholesterol, and craigsterol and hasidsterol. Yeah. All right, we'll move over to the other side. So this table, if you're paying attention, which parasite causes the most severe form of malaria? Exactly. Easy peasy. They do all cause malaria though, note that. Next table, which of the following is not an example of innate immunity? Yeah, correct. Easy. Yeah, low pH is acidic, high pH is alkaline. All right, next table. Actions of the complement system include, a lot of reading. See? So that's correct. Remember, joining forms to cause them to lyse and die is the membrane attack complex, and binding to bacterial cell walls to make them more recognizable is opsonization. This remote sucks. Okay. So an infected cut has become swollen and is leaking a white fluid. The fluid is most likely constituted of neutrophils. That's good because they are the first and main phagocytic cells that die once they eat too much. All right, this is a long one, so I'll pick a big table. This one. Two correct statements. CNF. How did you read that quick? Jeez. Yes, CNF. So remember that gram-positive, they stain purple because they have the thick layer that can retain that dye. And then gram-negative, they stain pink because the thin layer doesn't retain the purple dye, so it can then be counter-stained by that pink dye. G'day, fellas. How you doing, Nathan? All right, next one. Which of these sites are normally sterile? We'll go over to the far table here, the big table. Oh, you guys can do these questions as well. Just keep it in your head. You know, good learn. Hey, come on. Huh? BC.
你好。
G'day. Ni hao. So yes, BC. Remember, more inside the body is sterile. So the lower respiratory tract is sterile because they've got macrophages, alveolar macrophages, and then the upper urinary tract is sterile because of that flushing, lavaging action of the urine. All right, which of the following is incorrect about the body's natural defense barriers? Who wants to volunteer for this one? Nah, too bad. Okay. What'd you say? A. Okay, so the actual answer is D. Normal flora can produce lactic acid. It's just tears contain lysosomes, not amylase. Remember, amylase is what digests your carbohydrates. You've got salivary amylase, yeah, lysozyme, sorry. Good answer. Nice try though. This one is a bit of reading. I'll just give you the answer, honestly. So moist heat sterilization, remember, is quicker at higher temperatures. Oh, no, okay, my bad. The higher the temperature, the shorter amount of time that you'll need to sterilize something. Makes sense. All right, next question. It's a long one. So someone is experiencing severe diarrhea and stools are greasy, then foul smelling. She remarks that she recently returned from a holiday in Bali. What is the most likely parasite to have infected the patient? Let's go. I've already done, you guys. Do you want to have a go or no? Yes, Giardia. Just because of the greasy and then smelly stools. That's the main feature. How many more do I have? Damn. Okay. Tommy is a heroin addict who has AIDS. He was found dead in his apartment, surrounded by the feces of his kitten. Pathology showed an abscess in his brain. What parasite most likely caused the infection? Damn, that's a sad way to go. D? Yes. Toxoplasma gondii. Which of the following is false regarding plasmids and their role in resistance transfer? No, it's too long. All right, so the answer is B. So it can only be transferred from one bacteria to another. Yeah. Because remember, we have the conjugation, the transduction, all those other ways to transmit, the three ways. Which of the following is false regarding antibiotic use? D. I don't even know. I haven't even read it. Oh, wait. Do you have the slide? I just realized you guys had the slides up. Okay. Okay. Easy. So best of luck for your exams. This was supposed to be the last presentation, so not best of luck yet. You'll get that after the final thing.
All right, I think that was a very good presentation. Now, we just want to gauge how attentive all of you are. Do you want to do like a question time, or would you rather just go to lunch now and then come back. Because we have a couple of questions that we're going to answer from your cohort, but they'll take like 15 minutes. So who wants to do lunch now instead of questions?
Lunch? Yucky.
Nice. Who wants to do questions instead of lunch? All right. Yeah. See, they're going to get good grades in their exams, and the rest of you are going to fail. But yeah, we'll go to lunch now. It's 12:20, so we'll come back here at 1. Okay, it's 12:20. We'll come back at 1, and then we might give you another break after the question. So just come back at 1, please. If anyone's got any questions about anything, feel free to come ask us during lunch. But other than that, enjoy.
Okay. Okay, so let's start with the innate immunity. So that's your first line of defense. And the table that you're most likely familiar with is one of these, comparing the innate system to the adaptive system. And some of the most important ones, immune system is instant, adaptive takes days, specificity, immune memory, the cells involved. I'm sure you guys are very confident with this already, but it's really not two completely separate columns. They're really working together in tandem in order to fight the condition. And the kind of the connection is what I want to go into today. So firstly, to get on you, the bacteria will need to go past your natural barriers, right? So that's your anatomical barriers. First is your skin, covering pretty much all of your body. But your body actually isn't as enclosed as well as you think. There's like 5 holes on your head, there's some more down there, but there's so many ways that the bacteria could enter. And if you imagine from your mouth all the way to the rectum, it's really just a continuous tube that's been folded inside. And all of that needs to be protected against. And that's just through your mucous membranes, which just traps bacteria. And you can see that the ciliate membranes of the ciliate cells of the, for example, the respiratory tract, just basically just lots of rowers that beat the bacteria that have been trapped in the mucus above. There's things like coughing, sneezing, basically getting rid of the pathogen from your body. Unfortunately, it goes to everyone else, but at least it's not on you anymore. There's less for it to fight. There's the antimicrobials, just the proteins, for example, that are in your system to prevent it ever getting into your body. For example, the fatty acid, the lactic acid on the skin just makes it a very hostile place for any bacteria or pathogen to settle at. And there's obviously your normal flora, which Alex talked about this morning. Competing with your pathogens is taking up so much space that no invasive bacteria finds it really difficult to get in. So that's the first step. But once it gets into you, what you need to kind of watch out for, what you need to kind of like the immune system now will be the things in your blood, right? Your immune cells. So just to give you an overview, it starts as the stem cells in your bone marrow, and there's 2 different lineages. One is the myeloid. So these are the cells that basically after they mature, stay within the blood, and the lymphoid cells, which hang around more in the lymphoid organs. So the red blood cell, it goes to mast cell, erythrocyte. We're going to talk about that later, but let's focus on the immune cells right now. The mast cell, basophils, monocytes, macrophages, they're all part of the myeloid progenitor, and the lymphoid cells are, they mature and they mainly hang out in the lymph. We'll talk about that more later as well. But for the innate immune system, before we can do anything, it first needs to recognize the pathogens. So there's 2 things you really going to know, the PAMs and your DAMs. And in your lectures, you're just seeing things like the 5 different toll-like receptors and the crap ton of things, and the molecules and the name. You don't know really any of that. Basically, just to know that when there's pathogens in your body can recognize it. And the things that it wants to recognize are the pathogen-associated molecular patterns. These are basically just fragments of pathogens, and they're uniquely on pathogens. For example, the flagellum, the tail, the LPS, the RNA. And you might be wondering why the pathogen can't just evolve that way, but because they're actually very necessary for the survival. So you can't, for example, a bacteria can't survive without its lipid membrane. And that's just a fragment that alerts our immune system that something is invading us. Other things that activates are the DAMs, the damage-associated molecular patterns. So when your cells are damaged, for example, if a cell just dies, you don't want it to hang out. You want it to be cleaned up by the macrophages. Or if there's a cell that has cancer, you also want it to just be cleaned up. And how your immune system gets alerted to that is the DAMs, the damage-associated patterns. And both of that, they basically reach the pattern recognition receptors. So these can be intracellular, extracellular, really doesn't matter. You just need to know that this is how your immune cell is able to sample what's going on and know when to activate and when to not. So once it recognizes that with the PRR, there are the phagocytic cells that come to play. There's a cute picture of the macrophage. It just swallows. It can swallow hundreds. It's probably one of the best immune cells around. But what happens, there's 3 main types. The neutrophils, these are your almost suicide bombers. They kill and they die. And they're, when you get an infection, you see pus everywhere, right? That's basically what makes up most of it. The macrophages, it's a bit confusing what monocytes and macrophages have in different, but when they're in the blood, they're called macrophages, and they're called monocytes. And when they enter your tissue, they're macrophages. And on the bottom right corner, I think this is pretty cool that macrophages are everywhere and they're called different names depending on where they're at in the body. So you know the osteoclast, maybe learned about this already, but yeah, that's just also a form of of macrophages that just really resort the bones. And the last thing is the dendritic cells. So you might know them more as antigen presenting cells, but they actually also, they used to be macrophages, like they can phagocytose as well. So they basically just eat some pathogen and they digest it. They put them all over themselves, almost like decorating pieces of their body with the dead enemy. And they go around and activate the T cells. We'll talk about that later. And in terms of how it kills the bacteria after it phagocytoses it, this is the process, but essentially what happens is that it wraps it around, it finds that wraps it around, and the bacteria, the pathogen gets wrapped up, and it joins with a lysosome, which just contains a lot of like radicals, digestive enzymes, peroxide, just dangerous chemicals that destroy the genetic material and cause it to die. And yeah, the rest is just pretty much vomited out. And the other important part of the innate immune system cell are the natural killer cells. And the natural killer cells has two main components. There's the activating receptor, which tells the cell to kill the cell. And all cells have the ligand to that. So the natural killer cell naturally wants to go around and really killing everything it sees. That's why it's a natural killer cell. But luckily, there's also an inhibitory receptor on the natural killer cell. Otherwise, when it bumps around the cells around your blood, it's just going to kill all your good cells as well, right? And how it stops it is basically your target cells, your own cells. They've got the MHC class 1, which is expressed on all healthy cells, just to tell the natural cell, this natural killer cell, this is friend, it's not enemy, and you don't want to kill it. And one of the reasons the reason that natural killer cells are able to kill cancerous cells or viral infected cells is because the MHC class 1 is not functional. And when it's not functional, basically there's only the activating ligand telling the natural killer cell to kill without anything stopping it. And basically what happens is that natural killer cell will kill that. It will release lytic granules. Basically, again, just things like in the lysosomes, they're just very dangerous chemicals that will basically destroy the cell. And it just tells us to, kill itself, and it bears apart. This is the exact same mechanism as the CD8 cells and is part of the evolution, right? Why would we try to invent two different ways to kill a cell when there's a perfectly good mechanism right here? And another part of clinical relevance is in research into cancer, how this is applicable in cancer. So most cells, they have a lot of MHC class 1 because they're healthy. So the T killer cell, basically, the inhibitory signal overwhelms the activating signal. So it tells the natural killer cell that it's a healthy cell, don't kill. But what happens is that you can see that when it becomes virally infected or there's tumor, the cell knows to kind of turn down the MHC class 1, and it basically only tells the natural killer cell to kill, and that's how you die. The cells die. And yeah, it's basically like a balance between activating receptors and the MHC class 1. And what's really important about maybe cancer treatment in relation to this is that cancer, they often really smartly downregulate the activating ligand. So basically a natural killer cell, it wouldn't know to kill. And yeah, it's just some maybe application question that you guys will see, but it's really just a balance between the MHC class 1 and the activating ligand. And yeah, it's just a balance of signals. The other types of cells are granulocytes. So these guys don't come and phagocytose the pathogen. They basically release stuff. They're granules, right? That's why it's called the granulocytes. So N-eosinophil, they're the granulocytes that destroy helminth. Maybe this is semester 2, but this is just a parasitic infection because parasites, they're multicellular, right? They're way too big for a single macrophage to swallow. So you just release these stuff that kind of tries to kill them from the outside. There's the basophils. They mediate inflammation by releasing histamine. So this is also to do with allergy. And in people with allergy, it's really interesting to know that their granulocytes are activated. So these basically release way too much alarm signals when it's not really necessary. For example, when when you have hay fever, it just pollen enters your nose. There's nothing to attack it. It's nothing dangerous, but it releases histamines and just other immune pro-inflammatory mediators anyways, and that causes the immune reaction. And mast cells, yeah, it's also they're basically, you can consider them as very similar. They just release granules and that kills the pathogens. And this is probably a very high-yield summary of what inflammation is. So the 5 signs, I think it's pretty self-explanatory don't really need to memorize, but it's pretty self-explanatory, right? If you imagine cutting yourself and some dirt maybe got in, it will be red, it will be hot. Basically, that's because the blood is really flowing there to try to get the leukocytes in. They're swelling to make sure that you don't use that injured part of your body. There's loss of function just to make sure that you don't hurt yourself any further. So inflammation is legitimately very protective. I know that it might seem like a bad thing, but it's your immune system acting really quickly to make sure that you don't damage it any further and really just restricts the tissue damage to where the cell infection is. It's only when there's chronic inflammation that's really bad. And yeah, this is also something that really confused me earlier. It's just what's the difference between cytokines and chemokines? So what cytokines are, is basically signaling molecules that regulate the immune response. So it can be pro-inflammatory or anti-inflammatory. And there's so many. You can see the picture. It's like IL-1 all the way to like 30 or something. Again, it's probably not the most high-yield to know, but you just need to know that the reason that a T cell can go into all the different types of T helper cell is because of different immune cytokines telling what to do. Just basically ways to communicate between immune cells. And chemokines, they're a subtype of cytokines. So cytokines are just signaling molecules in between cells, right? That's all it is. Chemokines are like the breadcrumb. So they are cytokines that drive chemotaxis. Chemo means to do with chemistry. Taxis means move, like a taxi. So it's basically just allows the immune cell to follow a bunch of breadcrumbs to know where the cell infection is. Yeah, and in terms of how the leukocytes actually come is through this process. It's a zoom thing on top, but can't get rid of that. Okay, so the leukocyte, they're white blood cells, right? They just hang out in the blood, and when they're, what happens is that the bacteria, the macrophages, devour the bacteria, they realize that they need help, and they send the molecules to get the other monocytes to come rolling in to help out. And the process basically goes, first is the exposure, the PRR, the receptors need to come into contact with the molecular patterns of pathogens or damage associated. And the endothelial cells, they upregulate their integrin and selectin. That's basically the one you know. It catches the leukocyte, so it rolls, stops. And because of inflammation, the endothelial cells in the blood vessel is a lot more permeable, right? This is why you get swelling as well. It's a lot easier for the macrophages to squeeze through to come and help out. And this is a summary here. Yeah, probably worth remembering. There's usually a couple of questions on this. And the triple star, the selectin and the integrin are just the most important ways that your immune cells know to come, stop, and roll. And yeah, once it comes in, it just adheres to the wall and follows the chemokine gradient that we're talking about. So it really can pinpoint the site of injury. And under normal circumstances, obviously you don't want macrophages coming in to do all that damage. So the white blood cells, they basically just bind, they roll, and because there's no upregulation of the selectin integrin, they just keep rolling and go back to circulation. Despite the vascular permeability, just about inflammation. So you can see the normal blood vessel there, right? The endothelial cells are very tightly wrapping around your blood vessel, and that's good because you really don't want your blood vessels to be too leaky. But when there is inflammation, you want it to swell up. You want the mediators to come through. You want the white blood cells to roll in and clean up. So when it's acute inflammation, it opens it up a bit, increases the permeability, and you might be wondering why we need histamine if it's just for allergy response, but yeah, it is actually really helpful by the mast cells or basophils to make it easier for immune cells to come and clean up. And when there's chronic inflammation, you can imagine that the cells there's too much, it's always inflamed, there's way too much gap, there's too much swelling. And then when it is chronic, it's just really hard to return to the normal state. So things just keep leaking through, and that's why you get all that damage. The complement proteins, Alex already gone through today, but they're basically the molecular molecules. And from an immune perspective, the 3 things to know are just the 3 pathways activated. And we'll come back to this after we talk about antibodies, but just remember the classic, that's probably the most important, and the other ones are lectin. So lectin is just like sugars on the pathogen, and that's what your complement can bind to. We'll come back to this, but yeah, some questions to break it up. Is there a way to get rid of this Zoom thing? Can't really. All right. Oh. All right. But all right, so Simon went on a trip to, I think it was Africa, but when he came back, there's a bad case of a parasite. With a blood test, what cells will be a parasitic infection? Yeah, I think they saw. Did you guys see? Yeah, it's eosinophils. So these ones are, other ones are the neutrophils, macrophages. They're just for pathogens, for bacteria, and natural killer cells are for more viral infected cells. So yes, eosinophils, that's responsible for. Yeah, but next one you can. I just accidentally showed the answer. Okay.
Next one.
All right, which one of these is false? Raise your hand and here they will come over to you. Choose the table. Oh, okay. I think he found the victim already.
Owen was pretty confident.
I think it's D. Caspase is about apoptosis.
D then. We reckon it's D.
Let's have a look. E? Yep, it is right. The activation of caspase. So I put this question here is because sometimes they just throw random molecules, like who knows what IL-1 beta is in the caspase A, right? And a lot of times when there's multiple choice, if you just see a bunch of words that you never heard of, chances are it's probably not the right answer. It's usually the ones that you're familiar with because that's what faculty will be drumming home about, right? So the first one, PAMs are structures. Yep, so those lipopolysaccharides, double-strand DNA, RNA, that's just part of the pathogens. PAMs only exist on pathogens because it's literally pathogen-associated molecular patterns. Inflammasome is activating dying cells, and sometimes if you're not sure, you can just see if it makes sense, right? For example, dying cells will want to drive inflammation so it can be cleaned up by macrophages. So yeah. Which one of the following is... Is there a way to get rid of that?
Yeah, let me...
Innate immunity, I think. Innate immunity in your body is an example of innate immunity. Does that work?
We're going to choose this table over here.
E for elephant.
Yay.
Yeah, so I click E. No, I'll use this. Did something happen? Is it?
Now try.
Oh, okay, there we go. But yeah, don't confuse high pH with low pH. So low pH is obviously very acidic, and another thing. But yeah, you wouldn't want a high pH. If you have high pH, that's how you end up with stomach ulcers and lots of nasty infections. Okay, I think my clicker is working again. Okay, in an acute inflammation, neutrophils don't.
Got this table over here. What do you reckon? Yeah, I reckon. Do you want to?
No.
Calm. Okay, A.
All right. Yeah, so neutrophils don't. That's part of the granulocytes, the eosinophils and the mast cells. Nice. Okay. So that's it. Okay, so now we're talking about adaptive immunity. So this is where things actually get cool. These are kind of your special forces soldiers, right? You can imagine your innate immune system as basically just a local police guard. Then they got a baton, maybe they're not that good. If they can handle the easy stuff, but they can be overwhelmed really easily. And your adaptive immune system is the OP ones. They're special forces soldiers that have exactly the super weapon to counter a specific type of enemy. Now, adaptive is kind of a misleading word because adaptive suggests that you're actually adapting, right? But the T cells to counter that actually exist already. We'll talk about it in a second, but first what happens is that the immature dendritic cells, they have that shape so that they can really easily devour pathogens. And once it samples the pathogens, it grows the dendrites out. And the 3D one looks so much cooler. So it's like a little fluffy ball. But yeah, it just really increases surface area to contact the T helper cells. And what happens is that they leave the site of battle after they sample the enemy. They will go into the lymph nodes where the T cells hang out and find the exact match. And that's the key that is not really adaptive, more so specific immunity. Because in your lymph nodes, there's actually a T cell for every single possible type of pathogen that can ever exist and that will ever exist. Just through mixing the DNA, basically your body makes sure that there's really a library countering every single disease. So the role of the dendritic cell is just to go into the lymph vessel and just swim around basically until it comes into contact with the one that matches with the weapons that matches the pathogen. So once it finds that, it needs to activate with the major histocompatibility context. So remember the HSC plus MHC plus class 1 is present on all nucleated cells. It basically tells the natural killer cells that it's good, don't kill. But it can also present intracellular molecules. It's almost like a display window into what's happening for the cell because your immune cell is just roaming around. It obviously can't see what's inside of your cell. So it's almost like just a display window, right? So you can know what is going on there and just sample some things out. And when it samples MHC class 1, which presents a pathogen, like a viral fragment, for example, that is how it will, be activated and communicates with, so the dendritic cell with the MHC class 1, the MHC class 1 will communicate with the natural killer cell and the CD8 T cytotoxic cells. The class 2, they're also on the antigen presenting cells. And what happens is that they present the foreign material, which has been devoured. And what happens is that it communicates with the CD4 T helper cells. And to remember which one activates what, you can remember 1 times 8 is equal to 2 times 4, which is equal to 8. So MHC class 1 is to do with the CD8, MHC class 2 is to do with the CD4. And once it's being activated, how it works is that you can see there's an antigen presenting cell on the right there. So it activates both the CD8 cell through the MHC class 1 and the CD4 cell with the MHC class 2. And the other thing is that it requires co-stimulation. And now in the lecture, there might be 3 different molecules that can be the co-stimulatory molecule. That's not really that high yield. Just remember that there's a dual signal that needs to be activated. Because remember, what happens in autoimmune conditions is that your immune system goes into overdrive attacking something that's not there. The co-stimulation is basically the double factor authentication. It just really makes sure that the T cell is meant to be activated and not just going to the battlefield where no enemies exist and causing a bunch of damage to yourself. You can go and look up the co-stimulatory molecules if you want, but it's not that important. And the cell immunity, how it works is that, this T cell precursor, they are naive, and once it's being activated, it goes into the mature CD4 cell or the CD8 cell. And CD8 cell is same as natural killer. They just see if the molecule is cancerous or being virally infected and kills it. Otherwise, it leaves it alone. And the T helper cell is the one that's more interesting to us because this is really just a general of the whole army. Look at how many types of T helper cells there are, as well as all the different cytokines that it can secrete. So it's really specific in that only the specific T helper cell activates for different types of infections. So for example, there's a T helper cell 1 for, there's one for, can't go back, but there's one for intracellular, one for extracellular, there's one for viral, there's a different T cell for everything. And there's another one which is a T regulatory cell, and that's basically to help to tone down the immune response. And all of these ones, just remember that it's all the cytokines, the specific cytokines, maybe remember some big ones like the INF gamma, the tumor necrosis factor, but yeah, it's just know the big picture. So just an overview, right? The T cells are first mobilized, and when they encounter the dendritic cell, they basically bind to it, and it will go into either cytotoxic cells or the T helper cell. And really important how T cells are the T helper cells, the CD4 cells are really at the center of the entire immune response, because not only does it go to the cytotoxic cells to really just better versions of natural killer cells, but it can also go to be T helper cells, right, which secretes cytokines that attracts other macrophages and neutrophils. So it really loops back into innate immunity. It tells the macrophages to work harder. It basically makes them more energetic and more specific to the pathogen. And also it can become more T cells to really expand this kind of army. So without your CD4 cell, which is what happens when you have HIV, it's not just your an adaptive immune system that's kind of completely hamstrung, but it can't stimulate the innate immunity, which is really where the bulk of your soldiers are at, right? That is why it's just so dangerous when the CD4 cells aren't around. And also why it's so important is because it activates a humoral response, which is B cells. So these cells are basically antibody factories, right? They got the receptors that they can secrete out into the blood, and they do basically the same thing as similar to what complements do, just better. And how it works is that on the right diagram, it's a bit scuffed. I joined two together, but you can see that the antigen presenting cell, it goes to the T4 first. That's a green cell on the bottom. And the green cell, it basically goes around and activates a B cell as well. And note that the B cell already has the sample, like a sample of the antigen. It needs to be the same one just to make sure that it's supposed to be activated. And the reason this is so important to make sure that the antigen on the CD4 is the same as the one on the B cell is that you don't want it to be activating without reason, right? That's how allergies happen. It's just IgE immunoglobulins just there for no reason. So yeah, it finds the same one, and the B cell goes through what's called clonal expansion just to really rapidly divide to make as many antibodies as possible. And some of them become memory T cells as well. So that's how things like vaccines work. It's really hard to get infected by the same thing twice. Yeah, the clonal expansion basically produces a whole ton of plasma cells that secrete so many antibodies that's very specific, and memory B cells as well to make sure that you don't get reinfected as easily. So this is the 5 antibodies, and knowing how they work is actually pretty high yield. So I know that most of the other receptors and the different names aren't that important, but the 5 times are very high yield and almost certainly giving an exam. So the 5 of them are IgM, which is evolving the early response. IgG, that's the good one. Its passive immunity is in blood. It's the most common type. It's the one that comes later. IgA is in the mucosa. IgE is in the allergy response, and IgD is kind of the unknown one. And I have a pretty cool mnemonic here. Now, this is credit to Med School Bro. Not sure if you guys know him. So if it's cringe, I'm saying it's not my fault. Okay. IgE is responsible for, is it like what happens when there's allergy, right? How do you spell allergy? Allergy. Don't let any English students tell you otherwise. It's allergy with an E. That's how I remember IgE is for allergy, okay? Same principle, IgA, mucosa. So this is what happens when you're just basically making your mucosa tissues to make sure that any pathogen lands on your mucosa just get annihilated straight away. IgM is the first one that comes, right? That's why it's M. You go to your mum first when you're sick. So it's the first one that comes whenever there's any problems. And then after a while, after there's the T helper cell activation, the antibodies class switch to IgG. So this one's a bit of a stretch, but it's a good one in the greatest concentration. And it's also in the placenta. What noises do baby make? Goo goo gaga. IgG, right? So there's a lot of G's there. So this is probably the most important one to remember. Yeah, just it's the greatest concentration, right? It's the GG, and it's basically how babies, they come out, they haven't got any immune system yet. Their mother's IgG transferred through the placenta is what's responsible for keeping them protected from any infections. IgD, this is a relevant one, stands for I don't give a, you have to rearrange it a bit, but this is the one that doesn't really matter. Yeah. Okay, and going back just a couple slides. So now that you hopefully remember what each of them are, what it does is equally as important. So there's two ends to an antibody, right? There's first the Y tip, which is the antibody binding site. And this is a part that's very, very specific. This is a part that binds to the pathogen. So you can see from the left to the right, it just surrounds the pathogen so they can't really do anything, right? It needs those receptors to get into your cells and infect you. But when they are kind of all surrounded by antibodies, they can't really get around anymore. And that antibody binding site, the Y tip, is very, very good at binding to the pathogen. But the constant tip is very, very good for binding to the macrophages. So what happens is that after it's been surrounded, the macrophages are really attracted, just chemically attracted to the constant portion. So when it does that, it's basically, put a massive marker on the pathogen, and the macrophages and the just other phagocytosis cells, they know exactly where to go, and it binds to the constant region, and it's almost like a zipper, just zips it in, and yeah. Other one is that it activates the complement system. Again, it just can see how it just really drums up the innate immune system. So they're really linked. Don't consider innate immunity and adaptive immunity as just two different arms, right? They're really interlinked through the T helper cells and also this antibody system. It activates the cytotoxic cells again because the cytotoxic cells, they can bind to the constant portion at the bottom, so it knows exactly where to go and exactly what to attack. And also it blocks the receptors, to make sure that the cells the pathogens can't hijack our own receptors to get into your cell. I have to go through this again, but yeah, hopefully it makes it a bit easier remembering the antibodies. Okay. Complement proteins, this is what I said I'll come back to. Basically, another link between the innate immune system and the adaptive immune system is how antibodies, they also upregulate the complements, right? And it's G and M antibodies that's responsible for activating the complements. How remember that GM, it's a car maker, they make classical cars, classical pathway for activation of complements. Hopefully that helps. Okay, and these are just a different types of receptors. So T cell receptor, you can see, is a straight line. There's only one place it binds, and it's just really to, between cells to communicate, to activate something, to make sure it's supposed to be activated. And the B cell receptor, it's got that classic Y shape where it's the energy binding site is very good at binding to the pathogen, so it just surrounds it, and the constant site is just good for your own immune cells. And in terms of how it works, it's a variable region. Again, not that relevant, but it's just your immune system, your B cell basically just mixes up its DNA so that it can produce lots of different variable regions, so it can attack every single type of pathogen that can ever exist. Okay, this one, which antibody class can cross a placenta and protect neonates, such as babies, from infection? And after my mnemonic, I'll be disappointed if anyone gets this wrong. Anyone yell out? Yeah, IgG, right? Because Google Gaga, that's the baby one. Okay. Again, back to the clinical subjectivity. We know that CD4, why HIV is so deadly, is just because when it knocks out the CD4, both your immune system, your innate system, and your adaptive system is basically gone. And why blood marrow transplant is a cure for something like the Boyden bubble today is because the blood marrow is where your cells, they mature. That's how they develop, right? So he would have a mutation in his bone marrow that doesn't let him produce the white blood cells. And once you give a transplant of a healthy donor, you will be able to produce your own immune cells and have a chance against pathogens. Yeah, we talked about how allergy is caused by IgG, the mast cells is overreactive, and the autoimmune system, again, when there's not much regulation, the T helper cell, the T regulatory cells, responsible for toning down the immune attack. That's what happens, and your immune system basically just goes into overdrive. Okay, the next part is hematology. So This is the blood part of blood. So we talked about the immune part of blood, but the other cells in blood is what is probably more known for, the red blood cells, right? Okay, so that's blood. If I go to you and now draw a vial of blood, that's what's going to look like. There's liquid plus like all that stuff. But when I spin it around to really separate the components by weight, this is what it's going to look like. So most of it, just more than half with plasma, so that's a clear liquid. It transports things like water, protein, the metabolites, which is just waste products, and nutrients to the cell. And that's the whole point of your circulatory system, right? It's like a garbage collection system that gets rid of the cell waste and delivers things like glucose, oxygen, everything else needs to survive. The other part that's more interesting to us in this component is the formed elements. So these are the cells. So you can see that only less than 1% are our immune cells. So those are the platelets and white blood cells. And in the blood test, if you notice that there's too much of any of the blood cells, yeah, you will know that something's wrong and there's probably an infection somewhere. And again, knowing the immune system is so important, right? Because through which cells are activated, which cells are kind of in high numbers, we know what pathogen has probably infected the patient. And the red cells, they just are transporting, they make up the remaining 45% of your blood. Okay, let's go into each of them in particular. So this is hemopoiesis, basically the process of making blood. You can remember the right-hand side diagram, but essentially, you know, when you're prenatal, when you're still in the womb, it's not really that much bone marrow. It only starts to kick in around 4 months, because if you think about the tiny baby bones, there's no space inside to even make the blood. There's no even bone marrow. It's mostly in the liver, and when you're born, pretty much the bone marrow takes over completely, and you can see that it starts to decline. That's because you will just be making less and less as you age, and there's not much you can do about it. And the most high yield point to remember here are what it requires. So vitamin B12, iron, folic acid, yeah. So this is the diagram of the blood pathway. They all start in the bone marrow as the hemopoietic stem cells. They can differentiate into the myeloid, which is the blood cells, or the lymphoid, which are the lymph cells. But they can also obviously make more of the stem cells. And we talked about the common myeloid generators already, but the ones that we haven't talked about much is the erythroblast. So whenever you see blast in medicine, it's basically cells that produce stuff, right? So in this case, it just blasts red blood cells out. There's a fibroblast, there's an osteoblast, it just blasts materials, right? The megakaryoblast, mega because it's absolutely huge. Kary, do you remember karyotypes from the biochem? So basically the cell just gets really big because the nucleus divides, but the cytoplasm doesn't go into half. So it just keeps getting bigger and bigger, bigger. When it bursts, it becomes a platelets. On the other side, the immune cells we were talking about. So it goes to lymphoblasts, which blast lymph cells, and that can go into plasma cells, which is B cells, and lymphocytes, they're the T cells. And T and B, T is because they mature in the thymus, and B is just they stay in the bone marrow until they enter circulation and mature. Okay, so red blood cells, again, this is just a copy and pasted PSP slide, but you can just have a read. There's this, you know, to be flexible, increase surface area, there's no nucleus. And the relevance of that is because when there's no nucleus, you can't have any organelles, right? All those ones have been kind of ejected because it doesn't need organelles. All it needs to do is to swim around and transport the oxygen around your body. And they only survive for 120 days. That's actually pretty important because in when you're doing blood tests, you know that red blood cells survive for 120 days, and basically it's a marker of what's happening over the last 3 months. The damaged cells are basically removed in the spleen or liver. And because there's no organelles, there's no mitochondria. There's no mitochondria means that it can't have any aerobic metabolism, so it can only use glycolysis. And that will come into play in a second. But right now, the reason that they're rare is because they carry this hemoglobin, is protein with 4 globin chains. And the center contains the heme group, which is where the oxygen actually binds to, right? Each has 2 alpha, 2 non-alpha. And then there's a lot, like 270 million on each red blood cell. The next part is the 2,3-DPG, which that's probably the most higher stuff I'm going to talk about in a second, but just know that's what hemoglobin looks like. Okay, which substance, we'll have a quick break. Which substances are required for hemopoiesis? We had this slide like 2 slides ago. Did I hear A? Nah, it's actually C. Yeah, so A's, so this is the thing, right? Where this iron, vitamin B, folic acid, just really remember that. Yeah, this is like a question every year. But the erythropoietin, so even though basically that's in the kidney, that's like a hormone telling your bone marrow to make blood, is not actually a substance required. So it's not a building block. It's more of like a signal molecule to tell you that you need to make more blood. So even though, I guess, depends on wording, it's technically required, but it's not like a building block. So yeah, those are the 3 that's really important. All right, this curve, it's probably one of the most confusing curves in medicine so far, but this is really important. So what happens is that hemoglobin, how much it likes to bind to oxygen is different depending on how much oxygen there is, right? So if you have a look on the right-hand side, I'm not sure if I can use can you guys see this mouse pointer? Oh, it's very laggy. Okay, but towards the right-hand side, that's when there's a lot of oxygen, right? So when there's a lot of oxygen, it really wants to bind to the oxygen, which is what the y-axis are. Y-axis is just how much of it, how much of the hemoglobin is full of oxygen. So where in our body is there a lot of oxygen when there's a lot of partial pressure of oxygen? Well, that's in the lungs, right? Because you're just getting fresh oxygen into your blood. And when the partial pressure of oxygen is high, your hemoglobin basically really wants to be almost fully saturated with oxygen so that it can it can grab all the oxygen that your lung breathe in so it can transport it around. So it's got very high density, we say. And when your body has it looks better on the apple emojis, but what happens when there's not much oxygen in your body? Where does that happen? That's in your tissues, right? Because your tissues have just finished using the oxygen to do all the glycolysis and all that stuff. So when there's not much oxygen, your hemoglobin kind of changes shape. So it dumps all of the oxygen. You can see that the y-axis is really low because it doesn't want to bind to oxygen anymore. It wants to deliver all that oxygen to your blood, to your tissues. So it kicks off the oxygen so it can get used up by your cells. And what happens is that it holds on to carbon dioxide because when your muscles, there's a lot of carbon dioxide, but not much oxygen. So it wants to bind to carbon dioxide so it can travel back to the lungs so you can just breathe it out. In terms of the curve shifts, this is the 2,3-DPG. Hopefully not too traumatic with biochem this morning, but don't even know any of the steps for immunology anyways, but glucose, somehow, some way it makes 2,3-DPG. What 2,3-DPG is, you don't really need to know. It's just a molecule that's produced as a product of glycolysis, which remember is the only way that your red blood cell can make energy. So yeah, when there's a lot of glycolysis happening, your red blood cells will make a lot of 2,3-DPG. And why that's relevant, it will come into play in a second, but this is the curve shift stuff, right? There's a left shift and right shift, and really trying to don't memorize this, but basically just think about what's happening when there's right shift, right? For example, let's say we are at a partial pressure of 20. So that's at your tissue, there's not much oxygen. And your right shift, what happens is that when it's exercise, it shifts to the right. And if you look at the curve, right, the red is what's happening without the shift. So at a 20 partial pressure, it may be holding on to 20% of the oxygen. But after the right shift, you can see that the Y value goes down. So that means that it's kicking off just that extra 5-7% of oxygen. And remember, where that 20 partial pressure is will be at your tissues. So you really don't want any oxygen still binded, still stuck to the hemoglobin, right? You want to get rid of all of them so it can be used as fuel for your cells. So through the right shift, when it's exercise, it will shift to the right because you want more oxygen, right? You don't want it to be stuck on the red blood cell. So that's what happens. And the left shift is when your body kind of adapts to be more efficient, when you don't need much oxygen. When you're sleeping, you don't need all that much oxygen. So it shifts to the left. So what that means is that at the same partial pressure in your tissue, you can see the Y value is higher. So it's holding on to more of the oxygen rather than displacing it so it can be used by your cells. So it's just a bit of conservation. And what causes a right shift? Think exercise, right? When you're exercising, your body temperature will increase. You will have higher 2,3-DPG because 2,3-DPG is a byproduct of glycolysis. So when you're using a lot of glucose, you'll be increasing your byproduct, right? You'll produce lactic acid, your body will be more acidic, so it'll be increase of hydrogen. So all these stuff are a result of exercising, so it will shift to the right so you can get more efficient at delivering the oxygen. And the left shift is just basically everything else. This is iron metabolism. It's basically not the most interesting slide. The same amount of iron goes in and goes out. Most of it is recycled. You can read the slide later. Okay. Again, more clinical perspective is why we care when hemoglobin levels decrease. Well, hemoglobin is responsible for just transporting off the oxygen, right? And when there's less hemoglobin, there's basically less oxygen going around to your cells. And that's what happens in anemia. And again, back to why it's really important with vitamin B12, iron, and folate as a product is because if your patient presents with a reduction in any of these, you should think anemia. And that might be explaining why they're more tired, other symptoms. So yeah, it can either be caused of less production of red blood cells. So there's less dietary, less building blocks through less dietary intake. There could be bone marrow disorder, and the reason for that is because that's where the red blood cells are made, right? Or there could just be more destruction. So if you're bleeding a lot, certain medication will cause red blood cells to die, toxic to them. Again, inherited disorders. But yeah, really common sense, don't memorize the symptoms, but when you don't have much oxygen, you'll just feel more tired, you'll feel weaker, short of breath because you're trying to breathe more, and pale because there's less red blood cells and hemoglobin. And these are the thrombocyte. Okay, so the other types of cells in the blood, they're the platelet cells. So they're very small colors. And remember what I was saying with how they're made. Basically, they just keep dividing, right? There's so many nucleuses, some like mega carrier, like a lot of carries, I guess. But yeah, it just gets really big. When it bursts, all the fragments are what the platelets are. And they're the same as thrombocytes, but they're technically not really cells, they're just fragments. And the function is just to adhere, activate, aggregate all the hemostasis, means stops blood. And that makes sense. When you get a cut, you want these guys to jump to action. And the pathways and there's some abnormalities. Penia just means less of, themia means more. So yeah, all the vocabulary honestly make a lot of sense just looking. Okay, and what happens when you get an injury? Imagine you get a cut. You want to stop that bleeding ASAP, right? So what happens is that the platelets are the first that come to action. They hit some factors that's in your tissue to know that the wall has burst. So it just jumps into action and it just attracts more of itself. You know, when the platelets are getting past, they can no longer get past when there's platelets activated. So it just kind of sticks to the site. But the problem is, it's just a bunch of cells stuck together. It's not that strong. And we really need something that's more substantial. And what that is the coagulation cascade, the platelet plug, so the fibrin clot, which is basically, you can imagine, the first is just chucking down the sandbags to stop it temporarily. And the other part is just reinforcing with like chains to make sure your sandbags don't get blown away. The platelet ones are pretty self-explanatory. They just activate as they come. Yeah, so when there's damage, it exposes the extracellular matrix, which has a lot of collagen, and the high yield part is a von Willebrand factor, probably named after von Willebrand who discovered it. But yeah, it just activates it, releases, like, don't know, I don't know stuff, the different pathways, but it just causes the platelets to come together, and that's very fast. It just stops the bleeding. But it's not very strong. So what happens is that we need the coagulation cascade. And the very fast overview is that it activates secondary hemostasis, which is a fibrin clot. And the blood clot is basically a primary plaque plus a fibrin tying everything together. So let's have a look at this cascade. Just a high yield fact you might want to note down is it's all pretty much all produced in the liver. liver's the go-to organ. You will learn more about it next year, but it does everything. And vitamin K is really important to produce clotting factors 2, 7, 9, and 10. I'm not sure if you guys know, but in babies, they don't really have that much vitamin K when they're born. So we give them a shot to make sure that in case they bleed, they have what it takes to make the 2, 7, 9, 10 so they can stop themselves. Again, it's the two pathways. There's an intrinsic and extrinsic pathway. And intrinsic is more to do with blood vessel damage. And the second one is to do with tissue damage. But what was really confusing, I think was really confusing, is that when you burst a blood vessel, obviously the blood vessel will be damaged and the tissue will be damaged, right? You can't really have blood vessel damage without it leaking to the tissue, just like you can't have tissue damage without bursting a couple of blood vessels. So the reason they call intrinsic and extrinsic isn't actually because of in your blood vessel, outside of blood vessel. It's actually because of the test tube. So intrinsic means that your blood has all the components it needs to activate it. It's all in the blood. But the extrinsic is because it needs tissue factor from tissue damage. So you drop some into the blood vial and it will clot. So they're basically both required. So don't think of it as in one, you can get intrinsic without extrinsic, and you can't get extrinsic without intrinsic. Like you have both at the same time, and they merge to form the common pathway. So this is a cascade. And the reason that there is a cascade, like I'm not going to talk into too much about what the different molecules are. You can read to that, but it's a ton. And the reason not in order is because some geniuses decided that it should be named in the order of discovery rather than the order of like the process. And I'm sure that like this is going to be a pain for many students to come for like the next century. But yeah, but one, maybe the most important to know here is that the fibrin is number one, right? That's the most important thing. Discover first, makes sense. And what you need for that is a prothrombin. That's what activates it. The second one, and everything else is not really in a good order. Like there's some ways to memorize. For example, you can go see for the intrinsic pathway, it goes 12, 11, 9, 10. So just someone who's really bad at counting. Or the extrinsic pathway, it goes 7 to 10. Don't really have a clever way of, but yeah, if you guys have really clever ways to remember that, you can share it around. There's a lot of different ways, but honestly, just remember this for the exam and you're gonna forget it straight away and remember the next year and the next year, and it's a cycle. But what's really important is, I think, on the perspective, why there's a calculation cascade in the first place, right? Because what's happening is that your blood vessel is burst, so you really want to activate the clot straight away. And you can imagine, let's say we only have fibrin, right? As soon as, for example, fibrin comes into contact with things in your tissue, the damage factors, it activates. But that's really inefficient because it would be like a one-to-one, right? Let's say you're fibrin and your friends are fibrin, and you guys are trying to build a wall to stop the blood flowing. If you do it individually, that is really, really slow, right? If you do a brick each and then a brick each, that's really, really slow. The calculation cascade is almost like a way to make things exponentially faster. So what happens is that, for example, the factor 12, there's not much of it, but each factor 12 activates 5 factor 11, and each factor 11 activates like 5 factor 9, and then they go on and they really kind of cascade. That's why it's called a cascade, right? So that is why this is like, you're building the brick house, and rather than laying the bricks individually, you're calling 5 friends, and you're getting those 5 friends to call 5 more friends to help. And you can imagine that is so much faster than doing individually. So that's why we have all this complicated process. It's really your body's smart way to, make it quick. And you see that intrinsic pathway is quicker, right? So this is what happens first. And just, this is not really maybe a bit too much there for the exam, but when it activates the thrombin, it kind of goes, so extreme pathway is only 2 steps, it's really fast, but there's not much of it, right? Can you see how there's only like 5 multiplied by twice? It's only like to the power of 2. The intrinsic pathway is a lot more powerful. So after you get the extrinsic pathway, it goes to the thrombin, and the thrombin can actually go back to make the intrinsic pathway faster, not only activating the thrombin. So that's how your body stops bleeding really quickly. Another clinically important thing of thrombin is that, of the entire pathway, is that it's not like it goes from 12 to 11 and then becomes factor 9. they're just kind of like enzymes or cofactors. They activate different stuff. They don't actually get used up in the process. That's why it's not like a 1-to-1 ratio, right? Each factor 12 can activate so much of factor 11, so on and so forth. Okay. The clot formation, you can read that if you're really interested. Spam some flashcards before the exam and you'll be fine. After you form the clot, you don't want to stay, right? You want it to dissolve as soon as the bleeding has been, as soon as your blood vessel heals itself. Because you might imagine a big clot, it might be liable to break off, and then that's how you end up with a pulmonary embolism. So it goes through your veins into your heart, and your heart goes in your lungs, blocks your lungs, you can't breathe, and bad things happen. So we need to really quickly resolve the blood. So it's just as important to get rid of the clot as it is to form the clot. And it's a bunch of different molecules, but the ones that you should take into mind is a plasmin. Plasmin comes from a plasminogen. So ogen means that it generates the thing before, right? So plasminogen, it generates a plasmin. So this is responsible for breaking the fibrin into, again, different dimers. And another clinical relevant cool thing is that there's something, have you guys heard of the D-dimer test? If you watch like Grey's Anatomy or just medical TV shows, and be like, get me a D-dimer test ASAP, like stat. But what it is basically, it's a measure to see if you have a blood clot, right? For example, if you want to work out if a patient's condition is due to like a clot blocking a vessel, your D-dimers will be elevated, imagine, if there's been a clot. So that's how you can really quickly work out what's causing, if it's a blood clot that's causing your problems. The function test, so pre-thrombin activated. The cool mnemonic I read was that there's thrombin, there's two tests. I think it's so there's an activated test and the PT test. One of the extrinsic ones is intrinsic. So intrinsic one is a TT one, right? That's table tennis. You play table tennis inside. The other one is just 1T because it's tennis. You play tennis outside, extrinsic. So just like maybe something to note in the future is that to remember this, like the weirder your mnemonics are, the easier it is for you. So just think of some whack stuff and know how to stick into your mind a lot easier. I think that's okay. So this is the disorders. Again, the reason that we study the biology is because when things go wrong, you know what's going on. The purpura is palpable discoloration. It's probably an issue with clotting. Your blood isn't getting stuck in the blood vessel. It's leaking out everywhere. You can read that in a second, but not the most high osmolar for interest. The one that is so important is the Virchow's triad. So these are basically the three classical risk factors of thrombosis. So thrombosis is basically a blood clot that's not been broken, not being cleaved up, and that's traveling around your body. So you can imagine that if you have endothelial injury, right, that means that it's causing the platelets to aggregate and cause a clot. If there's abnormal blood flow, if blood isn't flowing as smoothly, it can't, it might just, still that will be more likely for clots to form. If there's any problems, the patient needs a problem with the coagulation cascade or the plasmin, that's a normal coagulation. And when you have those three risk factors and you see with a patient that comes in maybe with like shortness of breath or something, always think very high risk of thrombosis. And the thrombosis, again, it goes into the lungs and gets stuck into the capillary of the vessels of the lungs. So basically that entire part of the lung isn't going to get any oxygen. So you'll just be, pretty much short a quarter of a lungness. Shortness of breath and other symptoms you can imagine. Next part, we'll run through this really quickly. It's a response to injury, but when your cells get injured, there's different things it can do. It can give up, just die, necrosis, apoptosis, or it can go back to a normal cell and recover, and that's what adaptation is. So the outcomes basically depends on the amount of damage, obviously the type of tissue. So some tissues are a lot better. So if you cut yourself on the skin, it will be fine in a couple of days. But if you get a cut in the heart, then that's never going to recover. So your heart, if there's any damage, will just forever be less competent. And some is permanent, like the brain. The duration is also important. If you that's the difference between acute inflammation and chronic, right? Acute is good. It's getting your body to fix it really quickly, getting all the helper cells to come in. And the chronic is when there's so much damage all the time, your normal cells can't heal, and that's how you end up with things like rheumatoid arthritis. Outcomes, it regenerates, that's completely fine. It adapts. So hypertrophy, for example, if you go to the gym, you're literally injuring your cells so that it knows to get bigger, right? That's an adaptation. There's hyperplasia, which is increasing numbers. There's atrophy, which is if you're not using like your body part anymore, the body will think it's not useful and just get rid of that, make it smaller. Metaplasia is when it changes. So meta just means change, right? So that's when it's no longer the same as it would be. And this could be a problem because it could lead to things like cancer. So cell death, there's two types. There's a normal pathway, there's a necrotic pathway. Normal is just when it tells it to, when your other cells tell to die, that's regulated. Your phagocytosis cells mop it up, recycling components, everything's fine. Necrosis is when it dies without you meaning to kill it. So that could be injury, that could be, for example, not enough oxygen, it just dies and it releases inflammatory content and that could just kind of spread like a wildfire. The types of necrosis I thought was pretty funny. It's the CC, like, did you guys go in the workshop? Was there like the really scary pictures of the forms of necrosis? But yeah, there's a cheese-like necrosis. The sponge-like of the brain. I didn't put images there, but you can search up if you want. Pretty disturbing. Wound healing. The important part is probably, so it's just putting everything together, right? Once you get a cut, you first want to stop the bleeding. That's number one priority. And then you inflame so that your immune cells come and clean up the damage. And then it proliferates, which is the healing part. And then the maturation part is just going back to normal. So first intention is the first thing you try is if it's a small cut, it just grows back together really nicely, like a clean surgical cut, or if you cut yourself with a knife, sharp knife, it will sort of clean, it will heal nicely. But second intention, you can, but yeah, it's just really bad because it no longer approximates and you're gonna have to need to use the fibroblast. So you can see that obviously this diagram is probably the best. There's just way too much damage for it to smoothly join up again. So there's a lot of fibrin, a huge clot. The fibroblast comes in. The fibroblasts are basically like the repair cells. And the problem is that it just blasts a lot of fibers, right? But the fibers aren't the same type of tissue as where the damage used to be before. So that's where you get a scar is no longer the same as before. You always see it if it's a large enough cut. Bone injury, pretty cool. This is the same thing. So not maybe this one mark on this, but exact same thing. It inflames, stops the blood first, and then bones heal a lot nicer. Some other question? Okay. Apoptosis is just normal physiological process, and necrosis is bad. So yeah, that's pretty much everything. Does anyone have any more questions before I hand off to
questions? Wow.
Awesome.
Damn. Well done. Round of applause, I reckon. Turn off the mic.
Say for like 5 and all.
All right. So, does anyone have any questions about any of the presentations we've had today, or about stuff that we didn't cover, like MedLaw or HKS, or did we hairpin someone? I think you do. Any questions? Try our best to answer them. It's good. Oh yeah, we're going. Yes.
Can you please explain which interleukins we need to know and like what each of them do?
James, mate, get into it.
To be completely honest though, it's just like how much you want to know, because there's like way too many to realistically know everything. If you're interested, know them. But even if you know, like for example, 10, I can't really give you 10 to know, right? Because it might be the other 2 that's on the exam. So yeah, to be honest, like I completely forgotten the interleukins. You don't it's just remember, forget until you use it every day in clinical practice, and you just remember forever. So just remember how much you want. Yeah, it could be pretty interesting, but it's just a lot to memorize.
I think that's a very good point. I was just going to say, don't worry about remembering specifics for the exam unless faculty points them out directly, because they're usually very nice when they make your exams anyway. It's about application rather than remembering.
So when we talk about the adaptive immunity, does the antigen needs to bind to the dendritic cell as well as the B cell to secrete the antibodies?
Another question for James.
What was the question, sorry?
So in the adaptive immunity, does the antigen needs to bind both to the dendritic as well as the B cell to secrete the antibodies?
Yeah, really good question. So the dendritic cell, they have the antigens, right? Because remember that they are the phagocytotic cells that eat the pathogen and displace antigens everywhere for the T cells. And the reason that B cells are antigen is because they just come into contact with the antigen that's already in the blood, and they kind of just put it back up to compare it with the T cells to see if it needs to activate. Because remember, the B cell will only kind of produce the IgG if the antigens on this cell matches with the one that the T cell is holding. So then the APCs, they get it by phagocytosis, but the B cell, they can just sample random bits and put it up. Yeah, so it doesn't eat it. Yeah.
Actually, I'm just waiting for the B cell to bind to the antigen till we can actually secrete the antibodies. So essentially, are we just waiting for the B cell to also bind to the same antigen as the dendritic cell to release, like secrete the antibodies?
Yeah, it's just to make sure that it's the same. But the reason it takes so long is because you can imagine the cell's tiny, right? You get so much lymph of your body. It just takes a while to find it. And yeah, there's a lot of processes to happen because there's literally millions of like all the T cells in your lymph nodes. Literally, I'm not kidding when it says it can defend against every possible pathogens that can ever possibly exist. So you can imagine how many it needs to bump through. That's why it takes so long until it finds the right one. And yeah.
Thank you.
Alrighty. Any more questions? Is that one over there, or is that? Damn, pretty smart. All right. Before we leave, we've got a couple of questions from your cohort regarding stuff, like general stuff about exams and the year level. So I'll just go through them pretty quickly. The first question is, what is the median or average mark for the Sem 1 exams last year? For those of you who don't know, they were 79% for the first one and 79.8 for the second one. If you want to know, the average for the bottom 1/3 was 67%. The average for the top 1/3 was 88.3 in the second exam. And in the first exam, it was 67 and 88. So pretty consistent across the two exams. This is the highest average you'll have in any of your exams across all of your years of doing med, because it's basically just year 12 bio with some added sprinkles. But don't worry, because the pass mark is 52% for the first one and 49.7 for the second one. So you are chilling. You'll be happy to know that faculty is very nice. If the majority of students in the cohort haven't answered the question correctly, they'll just take it out and not count it. So it was a bit unusual last time. Our first exam for Sem 1, all of the questions were included in the final count, but the second one, 94 out of the 100 questions were included. So they took out 6 questions. So if it's a bad question or everyone just doesn't know it, then they'll just take it out. So unless there's been like alternative things that have affected you throughout the semester, you should pass. Like it's not too bad. They don't hate you. How was our WAM calculated? So in your assessments tab, you would have seen that there's the 100 unit and the 200 unit. It's like in that table that they give you. Your WAM is calculated based on the stuff in your 200 unit. So none of your assignments matter. The only things that matter are your 4 exams throughout the year, and then your OCEs that you'll do later in the year, but you automatically get 100% for your OCEs as long as you pass them. So only 60% of your WAM is actually like from exams. And even then, everyone's WAM's really high because 40% is automatically 100%, but it doesn't actually count towards anything. At the end of the year, you'll get either a P or a F on your transcript. You won't get a transcript if you fail. So the only thing that can be on your transcript is a pass, right? No one will see your WAM. You will get a document from faculty with your WAM. You don't have to tell anyone if you don't want to. No one really cares.
Internal as well. The WAM is also internal, so it only applies to the medicine faculty. It's not a Monash University WAM as well.
Correct. So if you're got a scholarship with a minimum WAM or something, doesn't really matter because you'll retain it anyway. It's completely internal. It does not matter.
Do they use the internal WAM for scholarships or not?
No, I don't think I do. Again, it's impossible to get. We got a couple more. I'm sorry, we'll let you go. Does WAM matter for placement applications or internship applications? No, it doesn't. So every single year in med is the same thing as first year where the WAM does not matter. But in fourth and fifth year, if you do really well in your exams, you can ask faculty to make a document with your actual WAM on it, like your properly calculated WAM, and then you can submit that to your internship applications if you want. However, Victoria is the only state in the country that still has somewhat competitive applications. They got rid of Z scores, but they're still competitive. Don't believe me because I'm a second year med student, but I reckon by the time we graduate, they'll probably be like every other state and just be random allocation anyway. So there's nothing really to worry about. Just calm down, everyone. How much are we supposed to remember from pre-clin years for our final exam in year 5? You'll cover all your theory in your pre-clin years. However, there are no exams in year 5. Your final exams are in year 4. So in year 5, it's pretty chill. In your final exams in year 4, it's clinical application. So whatever you've learned in year 2, you should be covering all of the important stuff in clinical encounters in year 3 and 4 anyway. So again, there's really not much to worry about. So I think the key, there's some questions about placements as well. Placements depend on the year that you're in. In year 3, 4, and 5, you'll get no preferencing. They'll allocate you randomly, so you don't really get to choose. And they'll change every year as well. You change hospitals across the 3. I think the most important thing is your grades do not matter. You're in the only course where it literally does not matter. As long as you're passing and you're understanding what you're doing, it's fine. Your job is to become a doctor, not a walking Wikipedia dictionary. So as long as you know what you're doing, your grades don't matter. And good luck revising. If anyone's got any more questions that they want to share with the class, they can ask them now. But if you've got any more that you want to ask personally, just come up to us at the end. Thank you for these sessions.
Good luck, everyone.
Oh, wait, one more thing. I haven't organized these sessions. A bloke called Ramod has. He's a pre-clin VP. So I thought it would be nice that we could all say thank you to him on a video that we can send to him because he's put a lot of effort into revising all of these. So if I can just get it to work. All right, okay. On the count of 3, let's just say thank you, Ramod. 1, 2, 3. Thank you, Ramod. Thanks, bro. All right, that's about it. Thank you. Have a good swap back.